All right, welcome everyone today. Glad to see everybody back for the workshops. Today we're doing RNA workshop number 4, spatial RNA-seq, right? We've been really moving toward this, right? We started out with our bulk RNA-seq, which is kind of the workhorse, that people use for validation and kind of the stalwart old data sets. Then we moved into 2 lectures on single cell RNA-seq, right? Which has kind of really grown really famous and powerful since 2017 or so, and it's just becoming the foundation of a lot of different analysis, right, moving forward. And then finally, that's evolved to spatial transcriptomics, right? And this is kind of the new hottest field. I guess a couple of years ago, it's the hottest field. Now we're moving into digital biology. We'll have to talk about that later, but spatial transcriptomics is still really on the forefront, especially with the movement to things like Xenium and things like that, which we will talk about. So welcome. Today we're going to be basically splitting this in. Actually, we're not. This is part 1 and 2. I forget. So today we're doing basically starting at the counts and moving to mapping our spatial domains, like our tissue architecture, domain biology. So we're looking at the overview of everything. And for next time, our part 2 will be RNA Workshop 5, and we'll be going to do more of our deconvolution, our cell type analysis, and all of our downstream things that go with that. So great. So let's just get started here. As always, lately with these data sets, I'm just choosing a sample. I haven't read the paper. I don't know what it's going to be like. And the idea is, if you're given a random sample, what kind of analysis will you put toward it, and what will you do if things go bad or good, right? So that's what we're doing together. So just imagine, like, today we grab the tumor, you have some new data, and you and I are going to go through it together, and we'll see what we find, right? Just like before. So yeah, think about it. The progression here. So bulk RNA-seq had everything was just completely blended together and averaged together. So you didn't have a great resolution. You kind of just had what was going on over the whole deal, right? Where single cell ended up splitting it up, you were able to find clusters of heterogeneity within this data, right? So you had different cell types, things like that. But you didn't know where they were coming from, right? Where in the tissue does each cell actually sit? How does it associate with its neighbors? How was the tissue artifacts associated with this? Like, there's so many different things that the spatial brings to this, right? So, when we actually dissociated tumor down to single cell RNA, we end up losing that spatial context, right? The tumor edge, the immune-rich pocket, the necrotic core, the TLS structure. We just miss all that, right? We lose all of that. And while we do get the cell types and cell states, that's great, we lose this missing dimension that spatial transcriptomics can give to us, right? Instead of just associating the tissue, we're basically keeping it intact on the slide, and we're able to bring in and couple the location information with it, right? So yeah, like I mentioned, today we'll be going through from raw spots to domain analysis, and we'll do trajectory analysis and some cool things on the domains. And then workshop 5 we'll do more of the deconvolution to get you the cell type of each spot, and we'll move forward with the analysis with that. So why spatial in cancer anyway, right? Oh, we talked about this, right? Tumors are not just bags of cells, they're geographic ecosystems, right? The tumor core is hypoxic, it's metabolically distinct from the tumor edge. TLS can form in immune active regions. There's hot spots, cold spots, and none of this geography can survive the single cell dissociation, right? So you miss all of that. So this is what we're really getting out of this. So, if we had a bulk RNA-seq, we had like a 1 measurement per gene per sample, right? Single cell, we got it per cell. Now we're getting it per gene per spot, right? So, what we're measuring is like the average across cells, then per cell expression, now per pseudobulk expression, and I'll talk about that. And then we can do things, cool things like infer different things that spatial can give you over single cell RNA. See things like spatial structure of heterogeneity, things like that. So anyway, the key limitation is probably just the spot mixtures for Visium, which we'll talk about, and that's being resolved as we move forward anyway. But we'll talk about the advances of that. So what is our data for today? Well, we're going to use this Wu et al. 2021 Breast Cancer Atlas. It's a very landmark paper, and it is used in basically every recent spatial deconvolution method. This paper uses this. So it's very well curated data. and it's very well known in the field. So you can get a good feel if the references match, when you're doing your validation. So basically, these guys took 6 primary breast tumors. They used 10x Visium. That's what we're going to work with, and over 26 patients, right? And they had a mix of HER2 positive, ER positive, HER2 positive, and TNBC. So we figured, well, TNBC, right? Well, I've worked in TNBC in the past, and it's just a very interesting tumor. So out of these 3 choices, I think TNBC was the coolest choice for us. So this is the CID4465. And why did we choose this? Why was it good? Well, TNBC, as you may already know, is triple negative breast cancer, which is defined by what the tumor lacks, which is it does not express estrogen receptor or progesterone receptor, and it doesn't have HER2 amplification. And about 10 to 15% of cancers are TNBC. And these are very aggressive breast cancers sometimes. They have high recurrence rates, short time to relapse, and historically fewer targeted options than the other ones. The ER positive, for instance, has a lot of good treatments. TNBC doesn't. And because it lacks, because you can latch on and treat through these hormones, and TNBC doesn't have anything to latch on to. So it's hard to send stuff to get it, right? And really, the TNBC kind of depends more on its tumor microenvironment, where that could be the immune infiltrate, stromal reactions, hypoxia, metabolic reprogramming. These are all things that tend to make the breast cancer subtype more. This just tells you whether the spatial transcriminance is more informative into these kind of things, especially because one of the things that spatial works really good with is comparing like interfaces, like when the tumor runs up against some of this TME. So to sum it up, there's rich tumor microenvironment biology that's often found in TNBC. There's things like CAF regions, TLS structures, a lot of things we can kind of go in there and try to find. And we have a actual paired single-cell RNA-seq reference available for this patient, which is awesome. It's pretty much one of the best things you can have. So basically, they have taken the sample and did Visium with it, and also took part of the sample and did single-cell RNA-seq, found the cell types, and we're going to be able to use that as our reference. Doesn't get much better than that to have that reference to pull back from. Otherwise, you'd have to pull from like a random atlas or something. So this is from our actually own data. So this is really good. So good tumor, good reference, and it's been benchmarked as a standard. This data set's been benchmarked and battle tested, and through cell chat, spatial domain method tests, trajectory analysis, whatever you can think of, they had it. So we can always go back after our analysis to this paper and really see how we did. So it's a really cool thing. So we feel like this is a really good tumor for us to grab. And this looks like it's a fresh frozen primary tumor, sectioned on the Visium slide. And let's see. We're going to use the same patient next time for the W5. And I guess we have some pathologist annotations for that too. We'll talk about that later. But let's move forward. I guess just the connection kind of for the analysis to the LAC workshops. Things like quality control, right? We normalized by DC2 size factors, right? In bulk, single-cell RNA, we did like the in feature, in counts, that kind of stuff. Well, that's going to be pulled over to Visium. Single-cell RNA, I'll just stick with the RNA one, the single-cell ones. We use SC transform, same thing. PCU map, same thing. Leighton, the same thing, plus some spatial stuff. Find variable features, find spatially variable features, right? Find all markers. A lot of similar things. So a lot of the things we learn in single-cell RNA-seq are easily ported over to spatial, right? They just have to, you have to just worry about the spatial context, right? And a lot of methods have been either it move forward or there's new methods coming that, are more spatially aware. But those methods are, there's too many of them right now, and it's very hard to tell which of them work. So it's better just to stay and learn from the basics. That's why I didn't bring in a bunch of crazy new methods, because there'll be another method tomorrow. And remember what I talked about, this is not engineering, this is painting. There's not a best method. You need to pull out from the ether which method to use and then try to validate them across your biology. And for different people, different methods will work better. And as these come out more and more, you'll have to use them more. But the idea is just the fundamental understanding of what's happening. If you have that, the rest of it's easy, right? So if you understood single-cell RNA-seq is just an extension, a spatially aware extension to the Visium data. So today, kind of our learning objectives, we're going to be loading the Space Ranger data, doing quality control on our SARAT spatial object we set up. We're going to do some more advanced principled QC, do our normalization. We're going to look for some spatially variable genes, do some clustering, find some Banksy spatial names, and we'll move forward and talk about some different things like gene module scoring, trajectory analysis, copy numbers. This is going to be a great lecture. Now, bear with me. This is a long one, right? I split this into 2 classes for the live workshop, so this is going to be a long one. So, you guys stick in, dig in. You can always stop it and come back later. So here are the packages I've loaded. You guys can look at every single thing I have used. Any new ones I used, I installed SARAT wrappers, which helps out with the Banksy, and I also installed Banksy. I use the legacy version because there were some issues with the newer one, and I also I wanted to make sure that different versions of R, people could use the same thing. So if you pull in the legacy ones, it works for all versions of R in reasonable time frame, and it's the same thing. There's not much difference. So I'm using the legacy Banksy, just so you know. And I'm making a quick little program, a little function here. It's a workshop helper. So it's kind of silly. In the new SARAT 5, when they output your spatial locations, the row names are the barcodes, and there's an X and a Y column, right, for the location. But for some silly reason, there's another column for barcode. But the barcode's already in the row names, so you don't need it. So it's kind of redundant, right? And when you try to put these things in cell chat, trajectory analysis, a bunch of stuff down the stream, it hates that and it throws errors. So all I've done is been a helper function, and it basically just returns X and Y. It just grabs that last column and gets rid of the last column, and all you're left with is row names of the samples, X and Y, and that works perfectly. So you'll see this if I use this get spot coordinates, all it's doing is just chopping down a thing. You also could just delete the last column if you wanted to, but I just kind of want to make it more automated. So before we get started on the analysis, let's talk a little about what Visium is, right? So the last time we were working with single cell RNA-seq, where we would capture, remember the cells in these little oil droplets, and that's how you could get one or maybe a doublet in there and be able to really increase this resolution. But this is interesting. Everything's moved. It's changed now. We've gone away from single cell droplets now, which was so famous in the field for a decade, and now we're just using barcodes again, matching barcodes that are pulled out, where you have oligo-DD probes where they can grab onto your RNA, right? So what you're doing now is we start off with actually putting the tissue inact on the slide itself, the tissue. You just get a good slice on it, fresh frozen FFPE, throw it on the slide, right? And you take a high resolution picture of that. So now, and you can stain it with different things, do IHC, you can do a lot of different things on it. I forget which ones you can do with the different things, but you can do a lot of different things. You get with a lot of different markers. So then you get like this picture already, this beautiful high dimensional picture of the actual tissue. It's already awesome enough, and you could go do analysis with. But then you permeabilize the slide, right? The DNA sinks down into these little hexagonal little shapes, right? And there's 4,992 of these little tiny little spots, and they're all barcoded with little of these barcoded probes. So what happens is when you permeabilize, it just sinks down into whatever spot it's in, and those probes capture the RNA. So now you have UMIs of all the transcripts across everything. But here's the downside. Each spot is 55 microns in diameter. That's too big for one cell. You could actually get between 1 and 2 cells, 10 cells in there. So even though it's a lot higher resolution, we still have a problem. It's really a mini bulk measurement. So there's always this like mixture of cells into that thing. Now, we want to do this 2 ways. So next time, in the next workshop, we're going to do deconvolution, which is difficult to do and a little shady sometimes, but we have a good data set, and this is when you can basically deconvolute each spot. And for each spot, you'll find the percentage of each cell type. And then you can kind of rank up the top one and be like, okay, this is the major cell type in this spot, whatever, right? So that's how you do that with Visium. The problem is, so we're going to do it today more at a domain level, which is much more applicable if you don't have the deconvolution results. And the next time when we have the more detailed results, we can show other methods and how they work. But don't fret. It's still a highly resolute highly resolute resolution, to be redundant about that. And okay, so this kind of just gives you a little bit of the background there. And where are we in the spatial transcriptomics landscape, right? So really, the Visium, the first Visium 1 and 2, let's go back even further. The 10x single cell RNA-seq, right? From 2017, that's when it first started, 16, 17, and then we could do single cell RNA, right? Then came along Visium version 1 and 2, and we had the 55 micron. This is what we're doing V2 now. And this is just becoming a very established workflow. It was at a good price point, and the price point stayed well as other technologies have gone higher. It's still well used, and you're often, like I said, you often are going to be given bulk RNA-seq data to work with. You're often going to give Visium data to work with. You're not going to get the most advanced data probably because it's very expensive to do those things, and only labs that have millions of dollars can really do those things, and they're more like methods papers, if anything. So Visium is perfectly, perfectly resolute to be able to attack a lot of really most biological problems right now. So don't feel like we're not doing better ones. This one's very good and well used. And if you can do this one, the others are easy. So Visium HD, same thing, but smaller resolution, right? We can capture one cell now. So really, it's the same exact analysis as now, except you don't have to do the deconvolution. Okay, so that's easy. We already learned the harder way. We can apply it the easier way. Slide-seq's kind of antiquated. It was really great before 2020, but it's kind of pulled off. There's a lot of resolution, but it's just difficult to use and expensive and really not. It's too meticulous to really work with. You have things like GeoMix DSP, which we'll reference later, but really this is really just about finding more ROIs on your spatial areas and comparing those along with IHC, Meriscope's another kind of example of that, and then we have Stereoseq 2 kind of advanced, but I will just point to Xenium here, which is the big brother of Visium HD, where we even have subcellular resolution now. So this is extremely cutting edge single cell imaging. You can do a lot cooler stuff with it. We can even match up protein subpanels, targeted gene panels, things like that. So this is if you're working in Nick Navin's lab and you have $1 million, you're going to pull that. But a lot of the other labs, you're going to end up having this kind of data. This is why I went back to this. So anyway, let's first load up the data, see what we get. We pull in the Xenadu records file from their paper, and we see with 3 files that we need here: the metadata file, spatial information, where it has like the actual picture and the locations, and a filtered count matrix tar GZ, right? This is going to have all of your count matrices, right? And this is the old style of doing it, and they split it into 3 files where they had just the count matrix, the barcode matrix, and the features matrix, and they kind of just, are CSVs, and they just kind of split them up into 3. And the new way of doing things, if you have Visium HD, it outputs, if you look below here, this filtered feature BC matrix.h5 file. This basically replaces this entire tar-z folder. If you're doing something in a brand new set, your directory would be set up like this: your sample, your filtered H5 matrix, and then a folder with all your spatial stuff. And then for this, it's really easy. You just do load 10x spatial, the path to the directory, the name of that matrix, and then your SA, pull it in. Really easy, right? So for us, we need to recreate that top folder because we have the bottom folder here, but we have that weird try thing. So we got to do something with that to make it work. Very easy. So first we're going to download. We're actually making a directory here, and we're actually just going to download the 3 files we talked about, unzip them. Here you go. They're all here unzipped. And we're just going to extract the tarballs, and then we're going to make them do some little data cleaning, and we're just going to kind of reorganizing them into a way we can kind of look at them in a sample directory that actually kind of makes sense. And if you see here, now we're in data woo, and I'll kind of show you what's happening here, like that data woo thing. Oops. You can see behind the scenes of my stuff here. Did I have data woo? Data. I guess I switched it back, didn't I? Apologize about that. So I forgot I don't have it in that directory. So it basically, show you like, what is going on? Let me look at that again. Now I'm like, these are the things where I'm like, wait a minute. There we go, data, right? I knew I had that folder. woo, and then here's everything, right? So this is a whole mess before we fixed it. And basically what I showed you there, that data cleaning, we basically just made these folders. See how these folders are all made? And then we have a feature barcode matrix and a spatial matrix. And you see how we have those three in here and the spatial ones here too. So in that way, it was organized in a way that it's they can be pulled in later, right? So you see here how we organized it, and we just put it in there. You could have just cut and pasted everything instead of having the complex code, because it's one sample, but this is the organization we need to pull in. And there's a little bit different to pull in, but it's really easy. You just do a 3-part deal here. Instead of that read 10x spatial, you have 3 parts. It's very easy. You do a read 10x on that filtered matrix, that pulls your counts in, read 10x image on that, just the sample directory of spatial, and then you put those together, your counts and spatial, into your object. Simple as that. And now you have our create object, and we're just basically going to filter the image here to the spots that actually exist in the matrix. We don't want spots that are outside. You know, they need to be inside the matrix, inside the actual tissue, because there'll be spots outside tissue. We want just spots inside tissue. All right, so great. So now we have our spatial object. We can go ahead and check that out and see what it looks like over here in RStudio. Where's our object? How do you spell? There we go. Oops, object. So now we have our assays. Remember, these are calculated later. I'm showing you the finished topic, but it's the same as before, the same as here at object, right? Metadata, all of our counts, everything. So it's just like we expect, same as before. No differences with the single cell RNA-seq. Like I said, it's not much different. And if you want to take a look at some of the columns, kind of peruse it, you can see it, but it's very similar to what you expect. Here's what I talked about if you use the get tissue coordinates thing, you get this nonsense here. And you see how there's a replicated column here? That's for my helper thing, knocked out this column. So you won't have to deal with that nonsense. And then it goes in just like this, so it works much better. Okay, so now we have the data loaded, right? And then we have everything set up to go in our object. So what's our first step always, right? Quality control, right? We've got to filter the data, quality control. But now we're doing a spatial quality control. And this is going to be different, right? And in this sense, the standard QC metrics aren't enough. And we're going to talk about why that is the case. So first, we're just going to pretend like this data is single-cell RNA-seq. And let's see what kind of filters we would apply. And then we'll come back around and see what we would do different with a little more spatial information. So the first thing we're going to do is just calculate our things. Remember, the calculator of mitochondrial percentage, add everything up and plot. Just like we did before, nothing different. And you see this real wide distribution in our features. That's something you're going to see more, even a pop up here. That makes a lot more sense because you have the 1 to 10 cells, right? So there's going to be more found and more distributed in the different places. And it's real tight, right? You're just looking at those very specific areas, very specific genes a lot of times. So especially with the Neiman things. So, and we have a low mitochondrial here. Remember, below 20 is always pretty solid. So we feel pretty about this data. Remember, we're looking at this as single cell RNA-seq now, even though the distributions are different. So we'll do what we always do. We'll cut the bottom here. I don't know, 1,000 or 500. We'll cut the bottom here, and then we'll knock this off at like, I don't know, 10. And then we'll knock this off, I don't know, 7,000 to kind of, because it really cuts up. We'll be a little bit strict here. It kind of cuts up there at the end. That could be nonsense. That could be doublets or something, right? So let's just stop there. Cut this off. Cut that thing off. Stop here around 4,000. Things like that, right? So what did I see? 507,000, 1,025. So 25, 30, 25. I cut that right there as it went up and cut the bottom a little bit. So you see here, greater than 500 or less than 7,000. Greater than 500 or less than 7,000. Greater than 1,000, less than 25,000. Okay? So we're just grabbing the main fat part of this guy and less than 10%, which is pretty good. That's a little bit strict because these guys could be something, but it's a good, solid rationale. All these things make a lot of sense for single cell RNA-seq, right? So things we want to talk about, look for next time in our spatial features. Well, do these flagged higher feature spots actually cluster spatially, or are they just scattered? That will tell us more about what's going on with those spots, if they're real or not. What about the actual counts, overall counts? Well, do high count spots differ? Do they form coherent structures? Are they spatial? Are they in right areas? Things like that. Those are the kind of things we're going to look at. Percent mitochondria. Do these things localize the tissue edges? That could be something like necrotic region or something like that. So really what we're trying to say is we need to look to the tissue now, to the actual biological, like pathology-based visual tissue, and also and just see if things pop out as we apply back our our counts on top of the data. We can kind of see if they fall in the right spots and if they make sense. So we're going to let the biology inform to verify our counts. So what do we see here? Just a little bit to start. What do we see as far as what the tissue is actually revealing? And I'm going to skip back 6.14. I'm going to go back and forth between these. Which one's easier to read for us? So I can zoom in on these really good. So again, genes detected per spot, okay? And UMI counts per spot, right? So percent mitochondrial, in-count spatial. So in-count spatial is showing a lot of stuff here, a lot of stuff here, some in here, popping up here. The scale's kind of off a little bit. It's kind of harder to see some of this stuff. And then the features, you see them up here and over here and over here. So what does that tell us? Does that make sense actually? Well, if you jump right off, it makes a lot of sense actually. The high count regions are in the really purple thick areas of the tumor, which are more likely to be tumor or just have a lot of things going on in there. So we see high counts in there. Where these pink regions that don't have much, it's more stromal or immune, they're going to have less counts. That makes sense too. Kind of same around this kind of area here, as opposed to kind of the more darkish purplish areas there. So that kind of makes sense over already. And mitochondrial, what's going on mitochondrial? Is it diffuse? So I don't really see many, just a couple a little here. All I see is this kind of line along here. But the way it is kind of this linear artifact moving along, it doesn't really feel like, to me, a necrotic thing. So what we're looking at is like coherence versus scatter, right? Tissue context, the light versus the purple, right? Remember, high count spots over dense purple regions are expected. Diffuse versus focal for mitochondrial. And what we see, that we see the biological pattern we expected. The dark, denser regions have the higher UMI, right? The lighter, more loosely organized regions are sparser. That's exactly what we'd expect, right? The low counts are sprinkled around, and the high counts at the top sit on visibly dense and tacked tumor regions with no linear streak. So it doesn't feel like there's going to be an artifact there. So if you have high counts, we might be worried, okay, there might be a tissue artifact there, right? Because that's for your doublet you're worrying about. But if you don't see that, you can be like, okay, those are just high counts. So do you see how we're informing our filtering and our QC based on the actual real biology? And of course, you'd be working with a pathologist one-on-one with this to get their information on this. Same with the the percent mitochondrial. We don't really have a coherent necrotic region. We just have that narrow spot across. This probably more going to be a tissue artifact with like cytoplasmic leakage during sectioning or permeabilization. That's probably going to be more likely what that is with the data we're seeing. And we can check that more later. So what we see is no obvious folds, no detached regions, and no low permeabilization patches, right? So we can feel like the filtering we have worked pretty well. And we can be pretty conservative and let a lot in because we're not seeing a lot of the tissue artifacts that would reflect bad quality cells. So we see here that the low count and low feature tails are mostly stromal biology. They're not artifact, right? And the lower bounds can be more lenient than the senile because of this, right? And the high count and high feature tails are actually in this dense purplish intact tumor, right? There's no folds or doublets. So these upper bounds can be dropped entirely. We can allow those cells because they're real cells, they're real biology. And the high mitochondrial, they're kind of scattered and stochastic. They're no big deal. We can leave those. They're not big problems. Let's just go ahead and relax all of these. We need to relax and walk back a lot of these thresholds, or they're just too strong. So we basically took off the top one here and just had a lower 200. We took off the top one here, moved that to 500, and then we moved mitochondrial up to 25%, right? And based on everything we see, this is a conservative and makes sense. Now, we're not going to stop here. We might find things later we don't like with artifacts and be able to knock them out and pull them out with different methods. This is just kind of a very hand-wavy way to do this, right? We need to do statistical methods to find other outliers. But this is a good way to start, and it kind of shows you the difference. Because remember, we're not looking for there's no doublets. there's 10 cells in one spot. Of course there's doublets, right? So we're not, in single cell RNA-seq, we're cutting the top off because they're most likely doublets. Here, we don't know what they are. They could be tissue artifact doublets, or they could be really high concentrated tissue. 10 cells overlapped in a very dense area. So anyway, what the interesting result here is, instead of our default filter that we had taking out 172 cells, now we only have one. So we basically did nothing. So we did all that work, and all we did was take out one cell. So it seems like we wasted our time, right? But actually, that's the entire point of what we did. We used tissue spatially informed biology to adjust the filtering parameters, right? And we would have thrown out 171 cells that are likely good cells that could be very interesting. So this did its job. Now, in other instances, we may find artifacts and throw out whole sections. It can go either way. The point is, you need to be validating these different things in spatial transcriptomics by multiple different orthogonal methods. That's the only way anything is going to be believable. I don't believe anything I see in these papers until I see those kind of things, because anything will give you a result. And there's so much parameter changing and filtering and normalization. Every paper, I mean, there's a lot of papers that aren't real. So you got to really, really triangulate these things. Sorry, I'm getting really animated, but if you want a good paper, and I want you to have a good paper, you need to go through and use multiple methods and multiple logic, combine biology, and combine the visual changes to be able to make these decisions. But anyway, this is not a failure. This is a success. This is just a really, really, really good sample. That's all you can say about it. It's a really clean sample. And we replot on here. We only took out a few cells, but the scale has changed a little bit. You can see a little bit here where we see the bigger cells, what we'd expect. And mitochondrial is the highest over here. And now a little more scattered because of the scale. But these are very things that make a lot of sense. I don't have any issue with keeping those in here. And really no tissue chunk is missing. No stroma region has been punched out. No hole. This is just clean data. Clean data, well filtered, ready to go. Just some things you can look for, just to let you know. You look for tissue folds, we just mentioned, right? Where these are like linear or curvilinear dark streaks where they have high in count and high in feature. And you can filter them based on if they're not good biology, or by validating with the actual histology. There's also these edge guys where they could have low in count and feature. You might want to filter, annotate these depending on what's happening there. You have necrotic regions, super important, where you have elevated mitochondrial content with reduced end feature, but also in a spatially coherent region. All those have to happen. Here you want to normally annotate these and kind of keep things there and kind of just filter when you're needed based on the analysis, because these are real biology. You want to keep it in there. And things like low permeabilization regions and detached things, you would definitely want to throw those out. Those are just not going to give you good results. Okay, so now we've kind of looked at things hand-wavy, right? We haven't added any statistics in there. So what if we want to add a third layer of validation, which was called principled spatial QC with local Moran's eye? And so far QC has been a combination of these standard global thresholds and visual H&E reading, right? To look for things like tissue artifacts, edge effects, things like that. But the problem is these global thresholds end up missing spatial outliers, right? Where a spot with 800 UMIs is fine in the sparse region, but it's kind of suspect in the dense regions. It's hard for global thresholds to figure that out, right? And our visual H&E readings by us and our pathologists is more of judgment-based and non-reproducible. If you see papers where a sample is given to multiple pathologists, you often get different results. So that's a problem too, right? So a third defensible layer is to use statistics, right? And principled spatial outlier detection, where each spot we're saying, is my QC metric anomalous compared to my spatial neighbors? Am I different, right? And this is exactly the same thing we're gonna use later when we look at TLS hotspots and hallmark spots. There's a lot of ways we're gonna be using this same statistical machinery, this local Moran's. It's an awesome, awesome way of doing things. And it should be, because this is the standard statistical measure for spatial autocorrelation. And where did they come from? Ecology. And isn't it so obvious that ecology is being mined for data and different kind of algos to be able to handle this data, right? Think about it. Ecology is basically spatial transcriptomics. It's really big. You have a bunch of animals around different areas, a different kind of DNA, different plants. There's spatial structure all around, this is literally spatial transcriptomics, but you're looking from space, right? So people have been borrowing a lot of ecology-based algorithms that have been really useful in spatial transcriptomics. And this Moran's eye has been one of them, right? And there's two ways it does this. It uses a global Moran's, which basically returns one number. And this summarizes the entire map, right? And it just basically saying, does the entire sample have good spatial structure? Or is it more random, right? And then local Moran's, which we did before, is looking to return one number per spot. So how looking at every spot, how spatially autocorrelated am I around to my neighbors to believe this actually is a real cluster of information, right? And this could be good or bad. We could find bad areas that need to be removed, or good areas that are interesting biology. That's the way you can use local Moran's to do these kind of things. And you can also do things if you have like when I was studying anthracosis in LUAD, like you could take an area that had anthracosis and one that didn't, and use local Moran's to see see the spatial structure of those two areas and see the autocorrelation of like anthracosis along with other different kind of cell types, things like that. That's the kind of things you could do it for. Okay, so the first thing we're going to do is use local Moran's, and really here's three places coming up we can use it. Is this spot's count low compared to its spatial neighbors? Is this spot's mitochondrial slot high compared to its neighbors? Or is there a tissue level gradient across the whole thing? This is just a few examples of the things we could do that would be cool, right? So let's first do the QC metrics, right? We are going to look first. So first thing to do with local Moran's. Now I'll tell you where local Moran's, you can run it inside CIRAD. There actually is a function for it. I'll show you down there a little bit lower, but it's messed up right now, and it's in a bug time where there needs to be a resolved bug. Right now I'll give you the wrong answer. So I'm going to show you a more a less sophisticated, conservative, by hand manual version of how to do this. And it works perfectly. You can always copy this code. And when CIRAD fixes that code, you can run it back in CIRAD. But it's going to be the same thing. So we're just going to get our coordinates. Remember with our function, so we just have our barcodes and their coordinates, and we're going to build spatial weights basically based on our nearest neighbors of 6, because that's the hexagon shape around me. And then I am going to run local Moran's on that total UMI count, looking around to my 6 neighbors, and I kind of just set it up here and run local Moran, and then I build a kind of matrix and show you what we're looking for here. So it basically returns 4 classes. We call high, high, low, low, high, low, and low, high. And what does this mean? So high means high spatial structure. Low means low spatial structure. So if you have high spatial structure and your neighbors do too, that's a good hotspot. Conversely, low and low means it's an anti-hotspot. There's not much going on there, right? And if you're high then low, that means that you're high relative to your neighbors. So you're probably going to be some kind of outlier, right? Your counts are too high, fold, who knows. Low high is the opposite. You're low, but your neighbors are high. You should be high. Something's wrong with you. So you're probably damaged or you have dropout. So that's a good way to know to look. So what we're going to do here is just grab those damaged ones. The rest of them we're going to leave, and we basically just pull out. We say, I want the low highs, and we pull those out instead of being 10 cells. Then we do the same thing on mitochondrial. We do local Moran here on our mitochondrial percentage to see what's going on with that. And here we want to flag the opposite, kind of like below, what we did at the very beginning, where we do high-low spots, where this spot is actually high while its neighbors are low. So that means that focal, spatial, mitochondrial percentage in kind of areas, that could be necrosis, right? So we want to get rid of those. Which this is kind of debatable here, because maybe we don't want to get rid of these because they could be necrosis. So we might want to flag these, but this is just showing you how you could get rid of them. Luckily there's 0, so it doesn't matter. And then we just kind of combine that. We end up getting rid of 10 cells completely, and we just kind of show on here where they were. It's kind of interesting. They're just kind of spaced out randomly, and that's good. If they were concentrated in a single zone, that might be that we're just discovering the global threshold that already existed, right? It's kind of trivial. So we just kind of found some guys that were high but around where they shouldn't be. We're able to get rid of those, and they kind of you saw where they're kind of on the edges there, which kind of makes sense because there's often biological processing things that can mess that kind of stuff up. Okay, so now we've done per sample. Let's do the whole tissue, okay? So we're going to use global Morans here. And the question is there a tissue-wide gradient or a tissue-wide spatial structure happening that could suggest an artifact? Because that would be a problem. So we basically do global Morans 1. We just do a Moran test on basically the logs. We got our count, our mitos, and our genes. And then we and go ahead and run on our special features here, and then kind of just plot it all up into a guide we can get. And it's basically just the same thing, just global instead of local. And we get a Moran's value. Pretty high, these two. So 0.415 is pretty moderate, no big deal. We already checked on H&E, so we know that's fine. These two guys are strong, which at face value looks scary, as something we maybe should look at a little closer, and obviously we should. But we already have. We've already looked at this. This is a very clean sample, and we already saw there was no artifacts. There's just no clear tissue folds. There's no clear rip aparts. You know what I mean? This is actually real spatial structure, and it makes sense because things like stromal and tumor and the things are separated really well. So there's strong spatial structure that actually is real. This is a perfect example of how it could have been bad, and this is why you have to go back to the actual real H&E with a biologist and go back and check this. I keep saying this, but you just got to, which is the whole point of having spatial stuff, right? And having it in the first place to add that to the value. So this kind of explains that a little bit below, but it's kind of pretty much the same thing I said. And here we kind of just give you kind of a summary, where the standard global thresholds just are great and a fast look at, but we need a little bit more to know what we did. We added on the visual reading. You always pair those two things. And then we added on some stats too. And when we were able to see things that looked good across the whole thing, global thresholds and pathologist H&E and principal outlier, in global Morans, if we think things that are orthogonal and we're seeing them over and over again, that's not redundant. That means they're actually right. And you start feeling things that are more believable. And one quick thing about just filtering in general I think is interesting. Just thinking about it theoretically, how much better filtering is in spatial. Whenever you do, especially bulk, but even single cell, you don't have any idea what's going on with the architecture. So you might get a bunch of T cells, for example, that seem good, yet they were on a tissue fold or something, and you just didn't know. How would you know? And they should have been adjusted or filtered or changed. And now you've reported a result that's been compromised, but there's no way you would have known. So that's an example of how spatial transcriptomics can just, even in one little aspect, really help to optimize, your QC. Okay, man, good progress. So we've nailed we've got our data, we've got our surrogate objects, we've done our filtering, all sorts of filtering, right? And everything's ready to go. So it's time to normalize, right? So now, because Visium spots, now we've been using S-tree transform already because it's already the top one to use. But we always said you could also use the log normalized. But with the Visium, it's not a good idea because of the mixture, right? Because of that variable number of cells, okay? The way that standard library size normalization goes, this handles it very poorly. It just treats the 8s, if there's 8 or 2 spots, it treats them the same. It can't account for this mixture. And it just ends up giving it kind of poor results. So in single cell spatial transcriptomics, definitely use S-tree transform and only use log normalize if a method specifically needs that. So we run this again, just like last time. Object, our assay, V2. We change the assay back. We call the assay SCT, and then we have our new SCT transformed assay with our 3,000 variable genes. And next steps, always do our PCA. We run PCA with full compotence. We do our elbow. We feel good about 30. We move along. We do our UMAC. We do our find nearest neighbors based on the 30. And then we do our Leighton clustering. We tried several different resolutions. And remember, choose 4 for Leighton. And your seed always. And look at the cluster tree. If you remember, as you go down, it's resolutions. These are the number of clusters and how they split. You kind of want to look for a good split before a bad split. So the top 2, just nothing much has happened. The third one, there's kind of been some splitting. I feel like this is decent. If you go one more, there's a lot of splitting happening. So that may be too much. So I decided to stop here at 0.6. It might be that too. I'm just painting a painting. How do I know? So I think this feels like a pretty good place to go. So we're choosing 0.6 and we're running our Leighton on that, and we're going to project our clusters back. There's our clusters, separate decently well. I don't like some of it. It probably would be worth going and playing around with Leighton for a little bit more on the second pass through to get a little bit better result. But for here, it doesn't really matter that much. You can see kind of the overlay here. It's not horrible. I mean, that's our stromal kind of area. That's our vascular area, our myeloid area, kind of. That's kind of our tumor area. That's our TLS area, maybe. I kind of already know things. So, but there's a lot. They're separating out things, but like, they just kind of seem patchy and blotchy, right? And they're mixed together in different domains and areas. We have overlap among different areas, and basically the thing is, when we do this, I mean, how is it supposed to do better? Leighton doesn't know where its neighbors are. It doesn't have the spatially aware information. It doesn't have like a prior that, oh, if a neighbor's closer to me, I'm probably more like him. It doesn't have that. It's totally just treating every cell as independent. So obviously you're going to get patchy things in transcriptional states that go to different domains. That's not necessarily wrong. Cell domains and cell types are different things. There might be different cell types within different domains. So this is something we'll address when we do our cell type analysis, next time, our next workshop. But for now, I'll go back to this guy. For now, we want to work on our spatially variable genes. So now we're doing pretty good. We have our tissue. We feel pretty great about it. Our clusters, span physically disconnected pieces, right? That's why I brought them together. But within the boundaries, they're more salt and pepper. And this just means a rough spatial signal, right? Edges are ragged and leaving, blah, blah. The upper core is a little bit messier, but this isn't a clustering failure. These are transcriptional states. They just didn't know where to put them, right? But this is why spatially aware methods actually exist, right? So there's lots of spatially aware methods for these kind of domain things, and there's one coming out every day. I think one just came out while I was talking, and who knows which one was right, which one was wrong. That's why I need you to know just the foundational knowledge that you can apply, and so you'll be able to apply to all these new methods and know what the foundational regions for why things happen, so you can do a better job of debugging your analysis, right? So we're going to work on finding spatially available genes, and then we'll talk more about Banksy and immune spatial aware clustering. So spatially variable genes, what does that even mean? What's the difference between a spatially variable gene and a highly variable gene? So far in bulk and single cell RNA, we've been looking at highly variable genes, and these are genes whose expression values vary more than expected across cells, right? And they're great for cell typing, clustering, things like that, because the cell types drive most of the variance. But that's not true in spatial, where a lot of the spatial geography drives the variance a lot of times. So we want to know which genes actually show non-random patterns in space, which ones are spatially variable, spatially highly variable, right? But you can have both. You can have a highly variable gene that's very randomly distributed, with no spatial structure, or you could have a gene that's moderate variance but has a highly concentrated region in one area. And basically the spatial ones are the most interesting for the most part. So you always want to look at those. But at the same time, you also want to look at the intersection between the high variance ones, because the intersection is a lot of times going to find important ones too. So let's go through that and try that out a little bit and compare and see what we actually get. So spatial correlation, right? So what does it measure, right? The variance for the spatial autocorrelation, right? Kind of variable tools that just kind of become spatially aware, right? So the first thing we're going to do is basically use the Moran's eye. Once again, and here's what I was talking about earlier, find spatially variable features method Moran's eye. So there's places where you can put Moran's eye in here like this, and they do work, but watch out for these bugs until they get resolved. Otherwise, you can use my methods. They're manual and they work great. So, as we talked about, Moran's eye is the classic measure of spatial autocorrelation, right? So for a given gene, it can be like, if I look at every spot's expression and compare it to the average expression of its neighbors, are there similar values clustered or scattered? So it's like, what's my spatial structure, right? Like, am I a gene that is around neighbors that are highly variable, right? And am I different enough, right? So to do that, we want to do Moran's I on actually our instead of the QC metrics, we're actually going to do these metrics on our gene expression data here. So we actually grab our spot coordinates and our actual features here. So we get our assay, we get our features. So we have our spots, our variable features, and then we're going to run our Moran's I across all of those guys, and we're going to basically get a Moran's I for all the genes. And when we do that, we find that there's 10 top. Then we plot the top 15 spatially variable genes using MRIs. These are all spatially variable. They can also be highly variable. We haven't filtered that out. And if you look a little bit closer here on some of these, we see some interesting stuff, like FTH1 is very localized in the kind of tumor admixture areas. Let's see, same with S100 over here. C24 is kind of same. This guy is kind of the opposite. It's kind of really low in those areas. IGSO2, kind of immune related, it looks like. IGSA, same thing. They're kind of located together. So if we look into this a little bit more, we see a couple things. We see plasma cell TLS biology. So if you look at like the IGSL2s, you're seeing a lot of activity up here that could likely be plasma related. Inflammatory myeloid. These guys, you see it over here. Tumor epithelial. We're kind of seeing it in kind of the areas with the more purple dense areas. So other guys are kind of housekeeping genes, and their spatial structure tracks cellularity rather than domains. They're kind of more separations than actually these domains we want to try to find. Anyway, so we're able to kind of find some spatially variable genes, plot them over, and kind of learn some interesting things about our different possible domains. That worked out really good. What's absent here? We knocked out a few here. Well, the problem here, some of these really important genes, they're also highly variable, and we purposely left those out. Or they didn't make the top of the list, right? Because they weren't spatially as strong as the other ones, but they're still really important, and they're there. So there's no reason to believe they're being left out. It actually makes sense why they're left out. Okay, so what about highly variable versus SVGO? So you can have both, right? You can have highly variable genes that aren't spatially variable, right? High variance, no spatial structure. You have the opposite. Spatial structure, but no with model variance. Both these things make sense, right? But what's cool, we talked about SVGs are probably the most important. SVGs, HVGs, intersections, probably the most important. SVGs by themselves, check them out. They might be important, but probably the least useful. So I did a few things. I grabbed HVG only here. I took from kind of the middle here and grabbed SVG only. It's a little bit of a hand wavy thing there. And I got some HVG only examples and some SVG only examples here. So that's really great. And we see HVG only, we see kind of scattered cell type markers. We see hemoglobin, capsaicin G, mast cell tryptase, different things like that. These are things that are present in the tissue but not organized. They're just kind of peppered around. They have high variance, which makes sense because a lot of cells don't have them. So if you have them, you're high variance, but they're not important. They're spread around. SVG only is kind of interesting. We see FN1. It's a fibronectin, which is broadly expressed across the stromal compartment. So that's going to be a moderate variance, right? But focally elevated. And then we see these guys are cellularity correlated. So the Moran's rule is the biology is not domain specific. They're just doing their job of cell typing, basically. But what are some really important ones down here that aren't there? Well, they live in the intersection. So remember, the intersection is often the place where you're going to find a lot of important things, where the SVG only is grabbing things like surface component markers, cellularity gradients, not novel focal niches, right? So you really want to look at all three, to be honest with you, just to see what you got. And here we're just kind of looking at the HVG only genes and the SVG only genes. You can take a look at those when you get a chance here. And we're kind of just seeing exactly what we talked about and would be looking. So really SVG is the one you trust the most. Then you go to the intersection, and then you go to HVG. But try them all. They're all important. So that works great for today. This is a very transparent way of doing this. It's a solid way of doing it, and it works great. If you want to go a little bit deeper to more rigorous method, you can use this NNSVG as a pretty good method, Spark X2. as SVGs, just a really good, easy R method to run. And while we did kind of our conceptually Moran's was good for here, but for actually a publication, something like NSMVG is a little bit more robust and statistically powerful. So what we did is we basically created our object, we turned it into a spatial experiment. We grabbed our Cartesian coordinates with our little helper function, created our spatial experiment object, log normalized, just kind of got the data ready, right? And then we filtered to the top 200 most variable genes, okay? And then we put those at a little jitter. This just helps out with the analysis. Don't worry about that. And then we run our NSVG here, right, with our log counts on our single cell object, and we get that, and lo and behold, we get a list of genes that are ranked up here at different values, and they're very similar ones. We're seeing the same ones. I mean, these are all the same ones we talked about. And when we compare the top of each one, the top 10 from each one, 5 out of the 10 are top 10. And that's just on a random ranking of log folding things. So we're getting a lot of overlap. That's good. The trick is to use multiple methods and show overlap. You would have been able to use NNSVG and then been like, we also did 2 other methods that showed these. Okay, that's how you get orthogonal validation that really matters and kicks butt for the reviewers, right? And then, so what's some cool things we found on this? When we did NSVG, what's different? Okay, here are the ones that are kind of the same, but like, let's look at some of the other interesting ones, right? It's kind of interesting, the differences. So NNSG promoted ribosomal proteins, we saw more of those, where Moran's kind of pushed them down. Moran's did more of the focal, that plasma biology, right? But it was harder for this method to pull that out. And CNS3 is very interesting. It's a very biologically novel difference. It's missed by Moran's completely. So we might want to check that out and see what it is, and let's learn about this. And this is the way you tackle this data. You find something interesting, let's jump on it. What could this be? Well, this gene is kappa casein. It's the most biologically novel NNSVG hit we just mentioned. It's a milk protein gene that makes lactational and luminal differentiation, right? Which is unusual for TNBC, but who knows? It's worth investigating. So we kind of looked at where it was. We saw a decent amount over here in the luminal, which makes sense, and the admixture tumor areas right here. So, this is a marker of luminal differentiation. Not what you expect, but, you have these mixtures sometimes. But what we really see is a broad distribution among both tumor compartments with substantial overlapping in these CD24 high tumor regions, right? So this is not like a peripheral ring, it's more of a tumor-wide signal. So the hypothesis could be, well, maybe this tumor carries like luminal differentiation despite its TNBC, right? That happens all the time. There's a large subtype specifically that does this, right? Where milk protein and luminal markers can be expressed together. And this is really important because it would triage you to a certain kind of treatment, right? But let's a little bit further. I want to know more about this. Like, is it really luminal? Let's look at the common luminal markers and see if they are over there as well too. So we make a luminal pattern panel here, and we look for it in the data, and it's rejected. We see nothing. There really is nothing going on here. KRD19 is the only thing really there, and nothing else is really there. And honestly, looking through the panel, AR is essentially a silent, which is the one we really suspect to see. Fox A1's not coherent. Guy3 is weak. KR18 is low. Really, KR19 is the only marker that broadly localizes with CSNN3. So really, the hypothesis, the LAR hypothesis is being rejected. We need to look a little closer to what actually is happening, because the expression is real. It's just not a subtype call. But what it is evidence of a retained luminal progenitor differentiation within what's otherwise a basal-like TNBC. And this is really cool. And there's a developmental theories behind this, and you can really look into this, and it could be a very interesting topic. So, closing that spatial variable function loophole there, 3 lessons we have from here, right? The method choice matters, right? It's very, very important, and we need to validate them. A single gene can generate a hypothesis, but we need a lot of markers to test it, right? And unbiased analysis in biology is not as great, but only if you can validate it, right? So remember, we keep going back to these multiple validations. It needs to happen. All right. So now we're going to do spatial domain integration. This is the conceptual centerpiece of workshop 4. So we've shown that standard graph-based clustering treats every spot as an independent observation, which is why our Leighton result was spatially fragmented, right? But spatially aware clustering adds to the prior that physically neighboring spots are usually similar, right? Seems pretty obvious. And there's a lot of new methods that are saying this. I'm choosing the most well-used ones in R, because the rest are probably Python, or likely Python. And we're going to try 2 different ones that are widely used and very powerful and are just as good as the rest. Remember, it's a painting. We don't know which one you got to use. You got to try different ones. So Banksy has this very cool idea where it basically is saying, instead of clustering each spot's own expression vector by itself, like Leighton does, let's cluster a vector that combines the spot's expression with the average expression of its neighbors. Now, I don't want to say it's just the average. It's a little more complicated than that. It also does this Gabor filter that creates this gradient. So really, it's kind of an average expression in the middle and a strong gradient on the edges, looking to cut off and find because you have to have a strong gradient to see where it ends, right? And to see areas that interact. So it has this average expression and this strong gradient around it, right? And it has basically one parameter, this random parameter from 0 to 1. The lowest, you get standard Leighton clustering, exactly like before. Point 2, you start adding on a little bit of areas, right? And you get, this is good for light neighborhood smoothing, like cell typing, right? But if you want to look for a neighborhood, you go a little bit higher, like 0.8. And it's grabbing things a lot bigger, so it's clustering huge areas together, right? And that makes sense, they'll stay together. The bigger the thing is, the more they'll stay together. So that's smoothing out to more multi-cell type domain regions, which is what we want today. Or we're more about the cell typing when we have the deconvolution. We're looking for the domain today because the mixtures are really good for finding domains. So let's jump in there. So one of the parameters also is KGM, and that's just because of the hexagonal grid wrong, the six things around it. That's why that 6 exists. And we're basically going to go through, if you want to use a second order one, I'm not doing it here, you can do 18, because there's the 18 around the second layer. But we're just going to stick with 6 now and try these 3 different values and see what happens. So here we go. We're going to run it Banksy through Seurat wrappers here. And we are going to, there's one little trick here to move around here, the Seurat 5. You basically have to get your coordinates and then create a new column here and then just say those coordinates to that column. We just need to create these new 2 columns. That's all it is. Basically splitting these coordinates to these 2 columns. And then we put those in here. It doesn't like grabbing that normal metadata column. I know it's annoying, but it's the way it is. And we have our lambda, our coordinates, our SET, everything else, our Geo6. Let me go ahead and run that. And then we do our find neighbors, clusters, and things like that. And then we go ahead and plot that here. And we see this is 0.2. And if you remember the Leighton clustering, it looks very similar to the Leighton clustering, actually, almost exactly the same as the Leighton clustering. Just a little bit more smoothed out a little bit. But there's still a lot of intermixing that we don't like here. But it could be, more of a cell type transitional state kind of issue. So now we're doing 0.8. We're doing exactly the same thing. And we're plotting here. And this is nice. We like this. I like this a lot. We're getting very smooth domains here. And, we're seeing this is all one domain, which it should be by itself. And we're seeing kind of, this is the purple area. It's by itself. Things are starting to plot out near the 2 purple areas are over here. And this is going to be kind of the lighter areas, so probably more tumor stromal. So these things are kind of already kind of making sense, and I kind of like how they look. And we can kind of look at it from a glance, and we can see how it just got better spatial structure. Here's the standard Leighton, very similar. It just had one more split up here. But the downside, the bottom one looks almost the same. So we didn't really get much advantage there, but here we get a lot more, because we have more coordinate awareness, right? And we have much more distinct regions here. And this is what we mean by spatial domains. The tumor mass is split across multiple domains to find different sub-regional structure. Now, you may ask, how are there more clusters? I thought we were going to find more domains, but we have more clusters. So this is not a bug, this is a feature. So in theory, yeah, you would expect less clusters, but if the biology demands more clusters, it will make them, and that's what we got there. So it definitely worked out really great, and we feel pretty good about that. So we got 8 different domains, right? So let's look at base space now. So base space is another really strong performer in independent benchmarks. You could also use it in R, and is established alternative and has better interpretability for some people because of the Bayes matter really helps interpretability. It basically takes a different mathematical approach. Instead of augmenting the expression and embedding like we talked about, it actually places an explicit Bayesian smoothness prior over a hidden Markov random field that kind of moves around, right, and samples cluster assignments via, MCMC, right? So the spatial coherence becomes the prior, not actually the embedding. So it's just kind of a different way of looking at it from different angles. So let's go ahead and run that and see what we get. So first we can just grab our spatial positions, plot those in, no big deal, same stuff. We build a single cell experiment object, that's what it likes. We run a little helper function that we know we like, and then we go ahead and process here. We put in our special object, we grab 15 PCs is the published value to use, they say, and we're using 2,000 of our HVGs here, and we'll see what we get. And one thing we like to look at is our likelihood plot here, and this kind of shows an elbow at 6, so we went with that, and we went ahead and ran everything, Q equals 6, Visium platform, top 15 PCAs, mclust, everything else looks good. And we want to run that, and go ahead and plot back, and here's what we get. This is kind of in the middle between the two, right? So they are smoothed out more. This looks kind of like tumor. That looks like that kind of area there. That looks like that stromal area. Maybe immune there. Maybe ECM. Maybe myeloid. Who the heck knows? But this got split a little bit more with the Leighton clustering. So maybe this should exist different because 8 said that. These two should not be different. That is a problem. These should probably be the same, most likely. So we're seeing a lot of similarities to the Banksy result. The Banksy result feels better biologically, but we're seeing a lot of overlap that makes me feel really good about both comparisons. And you can kind of look over them here and kind of see what I say. But you can kind of read through here and kind of seeing some of the differences and things. I mean, there's some things that base space is kind of more optimized for looking for, and things that Banksy is more optimized for looking for. So that's where you're going to get different shapes there, different granularity, simplicity. There's just a lot of different reasons why you would want to maybe look at these different ones. The best thing to do is try both, see which one fits the biology best. I think Banksy works the best for us, but we want validations. That's always important. And we have 3 right now, right? We have Leighton, Banksy, and base space. So we feel pretty good about where we're going with that. So now that we've annotated our actual we have our domains, now we need to actually annotate them, right? Annotated domains with mark regimes and H&E. So if let's say like we have our domains out, but we don't know what genes actually underlie the fundamental aspects of those domains, right? So the first thing to do is do find all markers and look at all of those, right? So we do find all markers, and we are going to find, just like single cell RNA-seq, look at all the other clusters, do Wilcox test, find the differential express change in each cluster. And then we did that, and we get our domain markers per each of our different, instead of clusters, now they're domains. One problem here that I want to talk about. If you remember this pseudo bulk thing I talked about in the single cell with a pseudo replication, how you couldn't, when you compared cross-contextually patients, you needed to do things like pseudo bulk aggregation and something like DSeq2 after. Well, this is where you have to start thinking about this in spatial concepts. So in a normal single cell RNA cell seq, where the variance was driven by cell type and we had no spatial structure, it was okay to use find all markers inside that. But things are different now. We're comparing different parts of the tumor that are completely different. Some have folds, some have necrosis. They're so different that they're like the different samples we talked about before. And they cannot be compared by this within final markers in cell, because you cannot treat the different regions that are being compared. You can't not treat them independent. They're not independent. So much has gone on in the area biologically, right? And this is the advantage of spatial, because you didn't have that level of granularity in single cell to be able to properly do that, right? So you were replicating probably within single cell RNA, you just didn't know it, and which is a big problem. So this is why spatial is just continually making it better, right? So what we're going to do here is, the difference here is we're doing, one difference I want to say here is, in theory, you can't do that. But what we're doing here is cell typing, okay? And this is a discovery based, and we're not going to take the p-value as the right value. We don't care what the p-value is, right? Everything's going to be pseudo-replicated. Everything's going to have a significant value because of the pseudo-replication. What we care about is the log2 fold change, right? So heck with that, they're all going to be significant. We just want to grab the ones that have the highest log fold change cluster. And for what we're doing, that makes sense. But we're not going to be like, those are the right ones, and that's it. We have to validate those, right? If we're going to take this approach that we know is a problem, then we can't just stand there at those are right. And you can kind of see here that sorting for key marker discovery, where we actually rank the candidates, then confirm against 2 orthogonal sources after that, then we can maybe believe they're true. And even then, we don't really know. So let's kind of look at those on the biology of the cells. And we kind of plot those out, and we're able to kind of tease out some domains here. C36, very strong here in this middle compartment. This guy's pretty spread out. HLC way over here, very interesting going on there. CL66, ATMM, blah, blah, pretty spread out in the tumor-like areas. IGF-1 in that kind of stromal-like area. So really seeing a lot of these just basic genes jump out and pick out areas. So what we're going to do is, just looking at all these different distributions of the expression of our genes, we're able to kind of hand-wavy come out with what we believe the domains are. And we come up with several different domains. An adipose vascular with C36, BAB4, RBP7. We have like an ECM remodeling stromal compartment with these guys. We have a plasma cell TLS hotspot, which is interesting. You saw that really high level up there. On that, we see a mixed stromal domain, apparent tumor differentiation, inflammatory myeloid, basal myeloepithelial, and a luminal tumor too. So we were able to do a pretty good job with these markers of being able to name out what these domains likely are, right? And when we look at 3 method conversions in the same biology, we're starting to see that some of this starts making sense here. So look, the TLS plasma, Moran's found that area to have these guys, ANSVG found it, and Banksy found it too. So we're seeing the same kind of thing across all 3. So we feel good about that domain. Inflammatory myeloid, same things. Moran's, NSVG, domain, luminal. We found that CNS3, remember, and the domain. So these domains are being cross-checked just in these little ways along the way. So now that we've done all this, it's time to go back to the H&E, like always, right? And we need to see things like, does the adipose domain overlap with light pink areas? That wouldn't make sense. Does the TLS domain overlap with strong purple aggregates? Does the inflammatory domain localize the regions with myeloinfiltration? Do the tumors overall on purple areas? Do the stromals are on boundaries are pink areas? So the domain biology should always be validated against histology. So we kind of felt good about that. We kind of labeled, based on everything we talked about, we made labels for each of our domain and plotted them. And looks pretty good. The TLS, the inflammatory myeloid, tumor are all falling on very dark purple areas. Stromal and vascular are on more lighter pink areas. Stromal ECM is kind of in the middle. Things are kind of making sense here. We feel pretty good about it, but there's one major mistake here. As we see in the middle here, is look at these kind of salmon dots here. So we originally said this is adipose vascular based on the CD36, FAB4, RBP7, APLNR. This is a textbook adipose signature. But also adipose tissue cannot exist in the center of this tumor. So the H&E is basically saying, even though that sounds right, it's not physically possible. So that can't be right. And basically what happened is a lot of TAMs, lipid handling TAMs, and different vascular guys share some of these markers. So it kind of tricked us into believing that. And this is what happens. It tricked us. We went back to H&E. We validated it. So this looks more like kind of a vascular lipid area, right? So we're going to relabel that, and it's going to be vascular lipid myeloid. So we're going to feel pretty good about that. So that's kind of our canonical lineage markers there. And then we'll go one step further and make a dot plot of very common markers that go around in each of those kind of cell types and domains. And then we're going to do one more validation and look at the expression of all these and see if they match. And if you go through here, and you'll see that like vascular myeloid is triple validated. A lot of these things line up and actually make a lot of sense. So we feel really, really good about that. So moving on a little farther, we're going to actually do a pathway analysis on that domain. So that differential expression analysis, so there's a pathways analysis on that actual differential expression between different guys. So what we're going to do is look at one guy. We're going to look at tumor. I tried TLS. Plasma was kind of a weird result. So I wanted a kind of simple result for you guys. So I chose one sample, tumor epithelial. That's it. And I grabbed the markers just for that. So we have all the markers, not the top 5. When we did that find all markers, now we have all the markers for luminal. We ranked them as always, and for our input. And then we also have hallmark gene sets that we pull in as well too. And based on that, we used FGCA and the ranked tumor markers and the hallmarks. We're able to find top hallmark pathways that are enriched in the tumor luminal domain. And they actually kind of make sense here. And we take the top enriched pathways and then we actually plot them. We're taking the very top one, the EMT. And the EMT is luminal, so we expect it to be there, but we're not finding it there. It's kind of diffuse in different places. So what could that actually mean? So we did everything right. But always when you look at the histology, things change, right? And the lesson here is pathways aren't always restricted to source domains. And that's informative in itself. So basically what's happening is there's this diffuse pattern, right? Where the score is elevated across the broader tumor and stromal, right? But also the strongest in the tumor luminal and visible also in the other places, right? So 2 effects probably produce this pattern. And it matters which is which, right? First, the hallmark EMT set is very promiscuous. It shares a lot of different genes with ECM, CAF, things. So it's going to light up a lot of different things. So that's some of your kind of contamination, the way you have to think about how you create these signals and what hallmarks you want to look at, right? So the other thing is real EMT-like programs do operate across multiple tumor subdomains. So there's this combination of some of them are real, some of them are artifacts, and it's hard to kind of get those out. So one way we can do, one way that's good is we added the pathway ranking, and we went from what genes distinguish just domain to what biology programs are active. So now we know what programs are active and where they're active. And that kind of tells us a little bit more than we wouldn't have had before. And also look at the other pathways. Remember the EMT, we had KRAS signaling up, myogenesis, apoptosis. We need to plot, in a real analysis, you would go and plot all the rest of the 4 of those, pull the whole story together, and honestly, it'd probably make a lot more sense. So let's project the rest of those. And now we see kind of here, apoptosis kind of all around this area, kind of interesting, spread out. KRAS is in that mixed myeloid section, but and a little bit in the tumor. EMT spread out. They're not great, but they're okay, right? So the EMT and the myogenesis co-localize, right? That's the finding, really. The ENS and myogenesis. EMT and myogenesis are these 2 right here. So these are the ones that kind of co-localize, and they show kind of a clear spatial structure. So this thing you can see, like myogenesis could possibly mean like a mesenchymal state here. It's hard to say. But since it spatially co-localizes, it probably does mean that. And then we're seeing KRAS signaling up, and apoptosis are real, but really subtle, and these are kind of diffuse patterns. And really, just showing you also domain is kind of a coherent EMT signal, right? So signals are real, but they're not spatially subtle. They're not giving us a lot of data, but they're giving us a bigger picture of what is going on. That's kind of the pros and cons of doing that. Not everything matters, but we put it all together, it matters. So closing that kind of subtype, we saw the SVG grab CS in3. We did multi-marker validation to kind of reject the hypothesis and learn a little bit more about a possible luminal progenitor retained state. we found some interesting things with Banksy and kind of looked at the pathway analysis and productions upon that. So once we've done that, what's the next step would be? Well, are these domains real? How do we know that? Well, pathologists, can't pathologists tell us? Well, lo and behold, yay, this actual, we need to compare our computational domains to a pathologist's expert annotations, and really multiple of them. And so the good news is this data set has pathologist annotations for every single tumor. So we're going to pull those out, load them, compare back, and do great. Wait, what? What is happening here? What is going on here? 73 stromal, 3 normal, and then you labeled everything else invasive cancer stromal lymphocytes? So this is crazy. Look at our side by side. They're like, that's normal stroma? Yeah, duh, we got that. And they're saying everything else is cancer. Well, great job, guys. That just solves every problem. Thank you for that. But so what's happening here is the pathologists don't know. So this is an awesome example. This tumor is so hard. It's going to be giving us so much trouble. But the best part of this is we're actually finding things that the pathologists couldn't figure out. Now, are we completely right about every one of them? No, but we're right about some of them. And we're learning information that's spatially resolved that a pathologist and even likely a computer vision computational pathology algorithm couldn't find either, because we need the genomic information too. The genomic information is adding, right? People are saying that it's going to be all visual, right? No, it's not going to be. You need this information. It adds on to it. And by having that initial genomic information, we're able to find these different domains that even the pathologist couldn't even try to do it. And then we compared, how much is compared to the actual to the real ones. They're obviously a horrible comparison because theirs are all wrong. But one thing we do see is very important is a 0.95 to stromal immune stromal. So the point is, every stromal one of ours matched their stromal. Every one of their things that wasn't some stupid mixture of everything we got right. So we got the ones that were right, that mattered, and we gave more to it and have triple validated it that it looks good. So we're even better than they are at that. And okay, we kind of hit a spot here. I'm gonna take a quick break since this is like a 3-hour lesson, and I'll be right back in 1 second. I'm just gonna let it run and just take a second to kind of just breathe in, think about what you're doing, think about what we've gone so far. Just take a breath, take a breather, and then we'll move forward. And this is kind of a good stopping point where we've hit the domains, we've validated them, and now we need to go a little farther. The next step is going to be doing copy number inference, interaction analysis, comprehensive module scoring, and trajectory analysis, as well as a multi-sample integration. So I'll be right back. Just take a second just to think about where we are, and then we'll keep moving forward here in a second. All right, we're back. That wasn't too bad, right? That wasn't too long. Quick break. Let's grab something to drink, take a break. It's hard for me. It's a lot for me to go. 3-hour long thing here, and we're halfway through, so we're making really good progress, I think, and we can pull through to the end. So just like in the last single cell RNA-seq, where we did our copy number inference, right, with copycat to find out, understand if our tumor is really aneuploid or not, because remember epithelial markers go between tumor and normal, so to be able to find something's aneuploidy, tumor is going to be, in comparison, usually have more genomic alterations, more copy number alterations than its colleagues in the different clusters and the immune and different things. So what you can often do is look for that aneuploidy, and that'll separate out your tumor. Then you'll know you'll have your tumor, which is very important in these analysis. And then plus you'll also have a copy number profile of your tumor that'll tell you a lot about the heterogeneity and different kind of biological programs and things happening in that tumor. It's like a gold mine, discovery, treasure chest. You pull up and find more information, right? And a lot of people don't do this, so it's very important. But always remember it's an inference, and don't take it as a grain of salt, right? And last time we used copycat. If you use copycat for spatial, it works too, but it takes forever, like days. So even on the HPC. So instead, we're going to use inverse CMV, which is very popular too. Those are the two most popular ones used. There's obviously newer ones too that work great too, and maybe even better. And they come out every day. Like I said, there'll be a better one next week. So just understand the basics of how this works and how to baby them, and then you can apply them and try different methods yourself. So, inverse CMV is pretty similar. It does a smooth gene expression across your jumping windows. It compares each spot to a reference population, and then it flags the differences, that have elevated gains or losses. But inverse CMV is also reference anchor design fits them pretty well because it understands these admixtures and ways to put in these different these domains pretty well. And you can put in those somewhat into copycat as well too. So it's not only for them, right? And really, this is the second orthogonal validation move for our Banksy domains. We've already said what the area is tumor. Pathologists have said that area is tumor. And then now we're going to say the markers have said it's tumor. And then now we're going to say the copycat says it's tumor. That will be the last step, right? So what we're going to do is choose our reference, right? That's going to be the hardest thing to do first. And as we saw last time, you have to baby these things along a lot. If you just put it in there, there's always this contamination. Last time we had a contamination of macrophages where they ate too many tumor cells, and copycat thought they were tumor. Those are the kind of things that happen. So the reference set is so important to set up to get these things done. There's two things. The reference set must be genuinely diploid. So if there's even an admixture of cells that throws everything off, you have to have a normal baseline to compare to, or the whole thing is useless. The other thing is the reference must be lineage representative. So you need to have a bunch of normal cells that have, a mixture of all the different cell types in the actual complete sample. So because what happens is if you have a diploid sample that does not contain a cell type, that sample is going to feel different to the algorithm, and it's going to say it's a copy number alteration, and we'll see a problem with that. So that takes away that problem, and you can't trust those values. So it's a reference. There's different things you can do. You can use the same patient but different areas, right? So that's what we're doing. We're saying we believe some areas are tumor, some areas are normal, and we're going to choose those as reference and tumoral as that goes. We could also use a paired match normal. That's the best possible thing, right? But you need an extra Visian sample, which is not always available, right? And we have that single cell data that could be a little bit helpful here, but really we need a normal. So the single cell data we have is more tumor, right? We need a normal. If we could pipe in normal into the sample, like introduce normal into the sample, cluster, normal will cluster by itself, and it would be very easy to set that up and do our comparison. So you can artificially put in normal cell of the same sample and do that, but it's always tricky. That stuff you always worry about. You're creating a problem when you don't need it. You could use an external reference, like we did with single R, where you use like an atlas, things like that. That's not quite as going to be good because there's always a problem with things not really matching. You really have to have a sample set reference that's just so like yours. You can also use sample mean, which I'm not even going to reference that because it's ridiculous to do that. But what we're going to do today is take different parts of our plots, our UMaps, and use those. So what we're going to do is anchor this whole thing on the pathologist normal confirmed stroma. So the pathologist said it was normal. We said it was stromal. We're going to go with that because we know it's right? And then looking at the markers for all the other cells, we see also vascular lipid myeloid has diploid markers, C68, 163, PKM1, has a lot of stuff that shows no aneuploidy. So we're gonna take a little bit of a risk here and combine these two guys together as our reference. And So we do have combined diploids in there, but they're not all in there. We did the best we could. They're going, so we have everything in there. We're going to go against everything else is going to be tested, including all the other Trumbull domains, everything else, right? So what are we going to do here? So we're going to set up our reference and observation groups here. And let's see. So we have we're grabbing our spatial counts here, and we're gonna do is so we grab our counts, really easy. First thing we do, immunoglobulin gene removal. So what happens here? These plasma cells express these immunoglobulin genes, and these often show up as artifacts and copy numbers. So we're just going to remove those so we won't have fake artifact values there. We're just grabbing our counts that have been filtered, and then our domain, remember, is the vascular lipid myeloid, and the reference, and then added on that reference set, right, of the stroma that we know are real. And we go ahead and use that, set them up as our reference. We set up our annotations. We set up our object like before, and then we go ahead and run the data. So we set up, our test spots, our reference spots, and then we go ahead and run those, create the object, and then we actually run the final SMB. It's kind of default parameters here. Things like this, like cells is better, but it takes longer. Use random trees, Leighton has a bug. These are just kind of standard things you want to keep. I6 is a more refined. They look at a lot of different kind of copy number changes. And then we basically just run this guy. It takes a long time. And then kind of here's one of our outputs. And it's kind of an output. These aren't basic names. These are actually the clusters of our different cells. Up here's our reference. So their diploid is pretty clean. So we feel pretty good about here. We also have something that's blank here. I bet that's part of that stromal immune population that we often have. And then among that, we see some copy number changes among all the cells, really. So really we expect to see them in just tumors. So we have to plot this back to see how this is actually working. But what's likely happening is kind of like copycat, that we have some artifacts and they're causing I guarantee that TLS section has artifacts that's causing fake copy number, even though we try to get around it. So we'll have to think about, like, maybe this is tumor because it's so strong, things like that. So the next step here is to plot this back on the graph and see what works the best, things like that. I'll leave that to you guys because we're so busy in this lecture. But this basically gives us, some chromosome 18 and 1 applications, which is kind of interesting. We're kind of missing application with MIC, which is kind of weird, but you never know what happened there. It could even be just something that happened in the sample prep, something to look at later. But the point is we have a clustered graph of different copy number annotations for our different regions, right? And what we see here is like chromosome 118, right? We're seeing that a lot, but we need to learn a little bit more about chromosome 18 because it's not canonical, right? And I'll let you guys read a little bit more about what we found here. We found some things that kind of made sense with like SMOD, and some things that didn't make sense with 8. And the diploid did a good job from aneuploid. The cluster we knew was normal. That's good. I think some other clusters you'd have to be like use the biology to believe if it's an artifact or real. And kind of like last time, as long as your normal's right, you can throw out all the other data and just look at the clusters, the tumor clusters alone, and just use those as the reference to that. And it doesn't account for any of the other ones. So those are real numbers. So when you overlay this back on your tumor, just use the tumor as your comparison back here as your value. So believe those values in the tumor references. The ones that aren't tumor, they could be artifact. Take those with a grain of salt, if that makes sense. Then we're just basically going to grab those files and visualize this actually on the tumor itself, where you see the copy number burden here, orange is higher, and we see it kind of in here, kind of in this tumor area, kind of this admixture area, kind of this area here, the plasma artifacts. HMM gives a little bit more resolution. Now we're seeing it a lot more in the tumor, in the luminal, in the TLRS, and this admixture. So those all make a lot of sense to actually where they are. So, feel pretty good about, where things are. One really interesting thing, let's do the cross-validation of these things. We're going to look at like all of our spots and see how well they do with their continuous HMM. And we'll kind of take a look at kind of the results here. And it's kind of we talked about the tumor aberrant at the bottom and had the highest copy number changes. The copy number instability is the most clonally advanced, possibly farthest down the tree, right? Inflammatory myeloid is kind of that admixture. It co-clusters with the tumor. Then we had TLS, but that was likely an artifact, not aneuploidy, based on the TLS biology. And then we had kind of a mid gradient here with basal myeloid, and we had kind of a low copy number, most differentiated tumor for the luminal, right? So, and then the stromal immune nailed it diploid. So all of these things in reference nailed it, right? So these things all kind of make a lot of sense. And I think we could go use those results, right? So the tumor gradient is real within lineages, right? The 3 epithelial tumor rankings, they go in order of what they should be, right? Same lineage, same reference contracts. So we feel a pretty good idea about their copy number heterogeneity. Where the 2 elevated non-tumor domains have opposite explanations, right? Inflammatory myeloid is a real tumor admixture. That makes sense why it has some stuff, but remember the TLS is more of an artifact, right? And some of the lower domains, they had low because they're low. So that kind of knocks out the copy number. You can check out the kind of the reserve there, and we're going to move on to comprehensive module scoring on tissue. So module scoring. Remember we talked about the add module score. We used that a few times in single cell RNA, and it works really good in single cell RNA. It works kind of good in spatial. Sometimes it works great, sometimes it doesn't. You gotta take it with a grain of salt. It's a lot harder. That admixture just kills it. But once we apply it next time to the deconvoluted spots, I bet it works a lot better. So what are the different things you can look for? Remember how we went through like a million things you can look for? Let's just remember a few of them and go ahead and apply them and see what we find here, okay? So we can do cell type signatures, cell state signatures, hallmark pathways, translational signatures. And if you remember about the module scoring, it basically works inside Seurat, and it just computes the mean expression. You give it a list of genes. It creates a gene signature, a custom gene signature. It looks at the mean expression of your genes minus the mean expression of size matched control genes from the same expression bins, right? So the score is near center, positive is more expressed, lower is less expressed. And it's just a really cool way to get quick results that you can look at later. The things you're going to look at, you're going to see like very dispersed, scattered. You'll see very focal hotspots. You'll see gradients. You'll see coherent regions. So these are the kind of things we're going to look at with our results to be able to go far, to be able to provide kind of an analysis or understanding. So the first thing we're going to do is cell type module scoring, which is kind of a no-no. We've done it here and there in the past, and it's worked some and not worked other places. It's definitely not the way you would want to do it. You want to use Banksy, you want to use deconvolution, but it's a quick way of doing it to get some pretty quick values of what could be right. And often it is right too, so it's hard to know. But what is important with Visium is because it's mixture, we're not telling, the result is not saying that a spot is a T cell. It's just saying this spot has an enrichment for T cell biology. It's just saying something T cell like is going on around there. You might want to look. That's all it's saying. It's not going any farther than that. And that's why you can imagine regional areas are kind of better captured by this. So you can make these, for instance, these cell type signatures I did for all these different one of our cell types we can find. And then we kind of, most of the genes were found in there, or really all of them. I kind of just plotted them out upon. And we're looking for cell types, and you see EMT is kind of pretty diffuse. Plasma is where it should be there, kind of weird there. CAFs are really high there where they should be. CAFs look actually pretty good. Endothelia looks pretty good. Macrophage, interesting, all there, the immune stromal interface. T cell scores in the T cell area, kind of interesting. And really, we have to kind of walk through them and see what we think. You know, that focal hotspot has that B plasma score. That's probably the most clean one. And it really shows, it really feels like there's some two brightly bounded hotspots that have the right cells and the genes involved with them. So like, really feel like with the gene signature we gave it, that's a pretty good idea about how to look out to find those. And that's probably our best example here. The other ones, you know, that's our focal hotspot, right? And coherent regions with macrophage, fibroblasts, we had coherent regions that we can kind of believe. The gene sets emphasizing that we see some kind of into like possible polarization gradient, or we have CAFs are just spatially shown across this part of the tumor, right? But really see that cell type signatures and spatial domain are different objects, right? A signature aggregates expression where its genes are switched on, regardless of which spot a domain was assigned. So it doesn't care about the domains, right? And that's why it doesn't have that spatially aware aspect. So there's some good things about it, some not things good about it. It's not good to cell type a whole area, but sometimes it's good to specialize little tiny clusters like we did with the T cells, if you remember. So this kind of just shows you a little bit of how I did the markers and how they worked for some without the others. And then finally we have that diffuse T cell score that's kind of just scattered everywhere. And a diffuse score is not a null result. It's saying that the cell type is distributed and not localized. And that's kind of what we have here. We have a lot of distributed non-localized markers. That's not saying they're false. That's just how our tumor is, right? And really, the downside of ab module score, you kind of look here at what I talked about, like it doesn't know how many cells, it doesn't know the admixture. So it makes some assumptions that easily could be wrong, and you have to kind of think about the biology in your question to see how well it is answering them. Because it's not useless, it's still very important, it's just tricky to use the correct way. And once we do the deconvolution, it'll work a lot better. And whenever you have a signature and you find a signature and it's working really well, you got to make sure, like, was that signature actually a signature or was it just one gene? So like we did in workshop 3, we ran our signature through to see what was going on, and we saw these guys are in none, these guys are in all, so these guys are useless. But then we saw some other things that tracked, through here, that nailed in the same place with the vascular. We're seeing some markers we're seeing enough markers that where the signature is real. And there's a pan-T markers kind of on the top here that kind of all converge on this together in the vascular, in the lipid myeloid and stromal immune, which are two immune-rich compartments. So really we're seeing multiple independent T cells lighting up in the same compartments, and the signature reflects real biology, not one gene. So this reliability retest that we always should do ends up actually passing. And we have some other interesting genes we can look at, and I'll give you a chance to look at this a little bit later. But it's not the best way of doing it, right? Next, just kind of compare some methods. We're using add module score that's inside Surat. There's another method called ucell that works almost exactly the same that's outside of Surat, and there's aucell2, which is not used that often. AUC is using the AUC recovery curve, and it is really good when you want to strict all signature genes top rate criterion, but not used as much. ucell, it uses a Mann-Whitney U statistic, so it's gene ranks, not raw expression like add module score is doing. It's a little bit more robust to dropout, which is really good, but it's a little bit slower, more computationally intensive, and it's really only useful on deconvolutions. Add module score is the only one that works well on mixtures. So when we deconvolute or we use different methods, we can use the ucell, but in that case, they actually work pretty similar. And once again, you can use both, and you can see which one's better. And like ucell, in theory, is better on sparse matrices, but the problem is the mixture throws it off because it doesn't know which gene should be on top. So anyway, we did some comparisons. See add module score here, add module score ucell is how you do that. We just wanted to compare a signature across a T cell score across the two. You see that the ucell one's very diffuse, while the Surat one's a lot more easier to see, right? The left one shows scattered T cell hotspots, where the UC cell is more uniformly dark. And this is not because the failure here is structural, or not because you have a bad gene list. Just think about it. UCell ranks genes within each spot, right? So the signature genes must be at the top to score, right? But Visium spots have lots of cells, and if other cells move to the top, like immune cells, housekeeping genes, it won't get scored that way, even though this thing's there. So that's how that ranking statistic makes it a little bit less sensitive than add module score. So I hope that makes sense. Next thing, spatial cell type scoring. Well, we've done that a lot. So remember, you have what? The cell types, right? Cell cycle. You have S, G2M, and G1, right? G2M is the growth, S is more the synthesis, G1 is the rest, right? So what we want to see is, do we see our spatial cycle artifacts, spatial saturated clustering artifacts within our data? And we talked about this before. If you have a cluster that is very bright, lighting up for cell cycling genes. It's separate, and it's very diverse. So that means it has a lot of different kind of cell types in it, right? Not just one. That is going to be cell cycling genes. And that makes sense because in a tumor, a lot of genes are cycling, this proliferation, right? So a lot of times a cell just is putting out all those proliferation genes. Those are just major genes. And it could be different kind of subtypes could all be doing that. So when we do our clustering, we're going to think those are one thing when they're not. They're all different cell types. They're all different domains. They're just all cycling. It's an artificial cluster. So we don't want that to happen. So how can we do that? The same way we did it before. Same way we did before. We just look at the genes. We grabbed our S and G2 genes, plotted them on here, and we see what's up. So S score, I see kind of spotty over here, spotty around the tumor area, luminal area. G2M, I don't see much. Don't see much there, right? And we looked at the G1 phases, G1 stores. There's about equal to those. So what I am seeing here is, so the opposite of this is G1, if you think about it. So G1 is probably scattered. It's kind of hard to say where G1 is because the relevance is so different. But I think what we're kind of seeing here is a modest proliferation with a tumor core lean in S phase. So our tumor core is leaning toward S phase, right? That's perfectly okay. It shows scattered warmth with a notable concentration in the lower piece, spanning the tumor apparent and the visceral myeloid domains, right? And the GUM is just kind of uniform. They're very scattered, low in magnitude, right? And what we really see here is we see a classic proliferated tumor concentrated in the tumor core. We see kind of concentration at leading edges, but we see it also diffuse throughout the tumor, which is actually bad because that means there's an aggressive proliferation throughout the whole tumor. That's actually worse than it being focal. So that's something you wouldn't be able to find out without spatial data. And then we also see an S phase lean, which is interesting, right? And G2M nearly absent, which is the moderate cycling fraction. So we're definitely not finding cell cycling artifact, something we should remove. We're finding real biology here. If we put it all together, it makes a lot of sense. Our tumor has a moderate copy number instability, retained luminal progenitor differentiation, EMT active rather than mitotically aggressive, and the S phase concentrations in the tumor apparent in the vascular lipid myeloid because it's consistent with a slow, spatially restricted proliferating subpopulation, cycling tumor cells and actively remodeling vasculature rather than globally cycling tissue. So this spatial distribution and proliferation is a recognizable prognostic feature in breast cancer. This is a real thing you can find. Saying a modest S lean in tumor and vascular myeloid compartments near absent GTM is a meaningful readout. That will lead to a proper diagnosis and treatment. So this is a real thing to be found. So that just shows you how cell cycling scoring can find a real important deal. So what about cell states, right? We did T cells. We looked at these last time in single cell RNA. So let's look at this in the spatial. So we have like our exhaustion, cytotoxicity, M1, M2 genes. We got all those signatures. We had the add module score, and then we went ahead and plotted them here. And what do we see? The top, we see the exhaustion and the cytotoxicity kind of overlapping a lot. Both just kind of warm scatter spots across everything, where it's just kind of like a mixed engaged but fatiguing state wherever they are. And then the macrophage polarization I thought would be a little bit starker. We're seeing a little bit more M2 over here, I believe, a little more M1 over here, but it's very hard to tell the difference. They're really broadly co-distributed, right? So we may see like a more of a mixed phenotype other than a straight gradient. So something further to look into on that, probably if you look farther into macrophages. And really, this is just showing us, this is a W4 answer to the same W3 question. We're just saying, we say neither T cells nor macrophages really segregate into spatially polarized states, right? The immune microenvironment is clearly co-distributed and unpolarized. And that's the finding, right? And single cell RNA could not have told us that. It just would have given us the average in states and expected these things to be there or not. It wouldn't give us the regional specificity to really understand what's going on in this biology. And that's the take-home message. You can go a little further and just look at the actual the cell states, if they're just a little bit more resolution on that, and you really are finding things around the tumor edge that where you'd expect them to be. And it's really just this mixed effector exhaustion pattern, to be honest with you, whether the programs are carried consistently. But what's interesting is this idea of, so single-cell RNA could not can identify exhaustion of the T cells, but they can't tell you where they are, right? And here it's pretty obvious that they're co-localized in a cytotoxic lean. So what's interesting about this is a mixed effector exhaustion state, which is actually consistent with a progenitor exhausted, it's called TPEX population. And this is a situation where the checkpoint blockade responsive subunit that retains effector capacity, right? It's consistent with this. That's what I'm trying to say. And confirming this identity, we'd have to go a little further to confirm it. This is actually something you can really look at because it changes the treatment in a huge way. So it's very prognostic to be able to know that. What else could we do? Well, we could do add module score just like we did before with hallmark signatures. So we did our hallmark pathways. They all had their own signatures, and we plotted them on the plots here to see what's up. Some of the ones we've already been doing. And we see EMT is the dominant one. It's across the stromal pattern. Hypoxia is scattered in both tissues. Glycosis oxfox a little bit different. I'll show you below. And the inflammatory stuff seems broadly distributed. So kind of the same things we've already been seeing. I'll let you look at these things a little bit closer. I can't go through every gene, every plot. I'll be here forever. And we do see a difference in oxphos, where we see oxphos in the tumor high down here, but we see the glycolysis is kind of more up here and up here. So we do see that difference in that, even though we have a negative spatial correlation here between these two. We're actually near zero, actually. So there's not much of it, not much zero. It's not much of a difference, but it's spatially different, right? Where we see glycolysis is active in two different compartments, in the stromal ECM and the inflammatory myeloid, right? And vascular lipid myeloid, where oxphos is in the stromal immune and tuber inherent, right? And this is just showing that glycolysis in immune and stromal compartments, and it's just showing that it runs glycolysis in immune and stromal compartments and oxphos in the tumor and stromal interface, metabolically compartmentalized, not switched, right? So the lesson is the spatial patterns that look inverse to the eye don't always survive a correlation test. So it looked obvious, but it's not surviving the test. But it's leaning that way, though, which is a biological way of doing it, right? Okay, so next, translational signatures. We're doing the same thing we always do. We make a signature, add module score, plot it, and see what's up. And we see here, we see both signatures are modest and broadly distributed. The TIS actually shows diffuse signature concentrated in the inflammatory myeloid and the lipid myeloid, right? Consistent with this IFN gamma inflammatory response in the hallmark panel, kind of similar to that, where the upper piece is pretty much dark, where the TLS score, very, very interesting, it's warm across the lower piece and right fragment, right? That's weird. And the upper piece is normally dark, but I thought TLS was actually up here. Which is very interesting. So what is happening here is the TLS score we're using is associated with the chemokine recruitment, not with the plasma aggregates. And this is really, really cool here. What we're seeing is we're seeing here that the TLS score we use captures the lymphocyte recruitment and signaling, where in here it captures the opposite would be the aggregates of it. So the opposite of that signal would be low here. They're the opposite of it. They've already grown and aggregated. They're very mature. So we're actually, that TLS signature is the opposite of what we thought, but it's finding what we wanted. So this is really cool. It's one more validation of that TLS signature, right? And the cool thing is here is a focal lymphoid aggregate adjacent to tumor, okay, with that TLS plasma cell, it predicts better anti-PD-1 response than diffuse low L signature. So the fact that we have that combination of mature, focal lymphoid niche and active chemokine recruitment, this could be a biologically favorable factor for the IC response. So another way we can look at this and connect back to the clinic and how important this is. We can also do spatial autocorrelation of module scores. So like our pathways, right? Which of our pathways themselves are most spatially structured? So we just looked at, we got our spatial weights and we ran the local neurons on actual global neurons on the pathways. And we see the global structure of these pathways here. So it gives us an understanding of what's going on there. EMT is always hitting the top, but it makes sense because these are spatially structured, right? Plasma is spatially structured. We know these are different. We know T cells are. So these are just really showing differences in how these things are structured, right? And it just shows that it separates strongly geographic from diffuse tumor programs. You can read a little bit more about this, but it's just saying, cell states don't form geographic niches. This confirms it. Okay, so cell states themselves aren't geographic niches, but they can have spatial structure within different areas. So it's the way to think about that. So think about it as Moran's eye ranks pathways by how much their topology depends on the geography, right? And the headline here is the tumor epithelial geography and the TLS niche are the two most spatially organized programs, followed by OXPHOS, right? And they all reflect real biology and identifiable spatial compartments, where the bottom tier is really kind of equally informative. We have an absence of spatial structure in cell state scores. It's really the quantitative version of what we saw earlier. Next up, let's look at local hotspots with global neurons. So let's going to grab on this to find out instead of the local things that are changing, right? If we have high, that's great. That means what we want: spatial structure, good spatial structure, good clustering. Low is going to be a negative cluster, kind of a cold zone. These are going to be outliers, and significance are good. We want to look for highs. So we run LISA just like we always have before, get our spot coordinates, run local Morans on our TLS score, because that's what we're looking at. And we see here all our different hotspots. We have 125 highs. Project back on the tissue, and this is really cool. I think it's a very good result. See the high here and the high here. We see the high here, low there, low there. You see the low there, low there. Nailed all of them, really good. And really what we're seeing here is really hot and low spots, right? We see 125 significant TL spots, 100 cold zones here, right? Overlapping this, and then we see, all these guys overlapping the inflammatory myeloid, and this guy is kind of on the edge of the tumor and the tumor immune interface here, right? So it basically sharpens that biology we talked about in 9B, where the upper piece is where the mature plasma cells concentrate, and the score is the highest, and it's also the cold zone. So that makes sense. It's the opposite of the score. It's the aggregates, right? And same with here. These were the ones that were getting recruitment signaling here, and so they're really jumping up and immune hot. So we're kind of immune hot, immune cold, and this is really, really great than just that continuous score, right? We're statistically showing regions are different, and now we can actually grab the genes, compare things like that. So this is just really great. It's upgrading visual and giving us straight statistics behind these different changes, right? Okay, so another thought to look at is our curated signatures, right? So we've been using curated data sets, like our choice. I choose my own custom signature or my own published signature, and I create the signature. We go look. That's one way of doing it. The alternate way is to discover modules from data-driven discovery, right? Use things like NMF to go out there and find the signatures that we should be looking for. That's the other way of doing that, which is perfectly awesome too. And that's what we will be doing, lo and behold, in our next workshop 5. So we'll get to that, just letting you know that I'm very aware of that, and we'll kind of move until then to be doing that. Okay, so now we're going to move to our next step, spatial domain inference analysis. And basically, the domains are so really important and really cool in the tumors, right? So what happens in these boundaries? You have the tumor stromal interfaces, the tumor immune interfaces, the stroma-stroma interfaces, and we find that the things that are going on in these interfaces are likely like the immune tumor interface. That's where the cool stuff's happening. That's where we want to find out and check on things. But how do we find those things? It's kind of complicated, right? Well, first you want to say if I'm in a domain, I would expect everyone around me to be the same, right? So what if one of you aren't? Or what if a couple of you aren't? Well, then you might be another domain. So if we go through every spot and say that, we can probably find spots that aren't really parts of the domain, or find, the interactions between domains. And I'll kind of show you some custom code I used to do that. And I was able to pull out all these really great interactions. It's like perfectly all these interactions, where you have this kind of a light tumor, immune here. We have immune tumor here. We have admixture tumor stromal here, tumor stromal. So these are all really good places to do interactions. So what I'm going to do really quick is I just want the tumor one. So I'm going to grab all of our tumor and just the interaction along the tumor here. You can kind of look into a little bit more on that. So basically, I want to go find the area. So I'll let you look at this. We're so low on time. I'm kind of going a little faster here, but we just want to know the difference between those interfaces, right? And so I just kind of grabbed it out and kind of grabbed out just the interface for the tumor. So you see here, just basically just did some data wrangling. Here is our tumor apparent structure, and here's every dot that's encompassing the top of it as a interaction dot. So what we're going to do is, this is going to be cool. It's low sample size, but we're going to do a differential gene expression between these two guys and see if we can find some cool genes that are up on the interface. That should be pretty cool, right? So we basically create a new identity, and we just say interface versus interior, grabs those two, and this is close enough to where the pseudo replication won't be that big of a problem. We do find all markers, and then here we go. These are differentially expressed genes that are up at the tumor stromal interface. Let's take a look at the genes here, and we see up at the interface the immune myeloid contact and matrix remodeling. The strongest hits are the myeloid immune, the LIL-RB3, which is a myeloid inhibitory receptor, NCF4, CST3. These are all transcriptive factors. This is a transcriptive factor tied to angiogenesis, immune cell function, invasion. There's other canonical genes for the tumor stromal boundary areas, such as TIMP1 and VIM. So we're catching up exactly the kind of things we would expect. So that's up at the interface. So what's up at the interior of the tumor, right? This is more CD4, GABRP, IGF-BB5. These are all epithelial luminal-based tumor identity things. That makes sense that they're moving toward that area. And there's some spillover to between, and that's going to have some interesting markers there as well. So now that we've seen that, kind of our differential genes between the two, we could do a like Hunnell plot or all the things we do. We have ranked genes. We could do whatever we want to do. I'm just going to do a quick hallmark pathways. I'm going to look between what's the difference in the hallmark pathways. I just found the interface, did the hallmark pathways, plotted them, and I saw the tumor stromal interface versus the interior. So the positive ones go toward the interface. We grabbed all these guys, and let's read it. We see a clean spatial difference here. Up at the interface, we see things like allograft regestion, interferon gamma response, interferon alpha response, and so on. These look toward, these are things that make sense that go up toward, the interface, right? To the immune engagement, EMT. Those are all the kind of things you expect at a tumor edge like that. When you look at the interior, it's more just like cell cycle, proliferation, things like that, tumor stuff, right? So we're seeing an obvious cool difference in those genes that are along the interaction. So I think this is really, really cool. We learned some really extra biology here. It shows you kind of the power of being able to do that. Our top signal is EMT once again, and we kind of just looked at it again because we wanted to see. What we basically did was we created a signature based on that edge score, right? And then EMT scored high. So we wanted to see how EMT scores on the invasive margin, and it was pretty much diffuse. It was broadly diffuse, and really, it's just saying that the edge is more an immune engagement zone. There isn't a straight ed cutoff. It's just where when you run together, there's just a lot of immunological engagement happening there, and it goes against what we talked about before. We have our tumor is moderate copy number stability, retain luminal differentiation. We have that TLS plasma niche. We're just the relatively differentiated immune warm TMBC. And it's really the kind is associated with checkpoint inhibitor responsiveness. This is really good information that the patient would be happy with. Exactly the kind of way why we're doing this, right? All right. So let's go to the last major analysis for today, spatial trajectory. And this is really the pseudotime is geographic distance. And remember before we did our spatial trajectory on our UMAP, and it kind of went through the different centroids of the clusters on the UMAP and was able to provide us kind of a biological gradient or continuum. When you do it in a spatial aspect, if there is no continuum, it'll just cluster out domains. If there is continuum or gradient, these are really important things to find. So you want to, especially when things move from the outside to the in, inside to the out, toward important areas. So we still want to look at this and be able to see these things. And things we want to care about are tumor evolution from edge to core, differentiation gradients in tumor cells, hypoxia gradients, immune infiltration depth. These are all the kind of things we can look for and try to, and it really, for these kind of things, you got to baby them along and come up with a very specific research question and then try again and again until it makes sense, until the biology actually makes sense. So the first thing we want to do, if we're doing a trajectory, it really can't go across pieces. So we want to just capture one piece right now. We use dbScan here. You guys can check this out, but I basically just grabbed the coordinates. I used dbScan on the coordinates, and I found these are the different ones. And I was able to grab out, cluster 3 here. So we have one sample to work with here. So that's what we did here. And then what we're going to do is we're going to look at, set this trajectory to go from the inflammatory myeloid over here all the way across to our tumor apparent. We're going to go here to here. So that's how we're going to anchor our trajectory. Remember, when I tried it alone, it picked some random thing. So we know this is the state that starts at the beginning of differentiation. We know this is the end. That's why we can feel good about our anchors. Now we use slingshot in single cell. Here we probably have to use a very complicated monocle or other Python-based things that would be very difficult for you to install. So I basically kind of just did this manually. It's not that hard. And you actually can just copy these 2 paragraphs and code blocks, and it can just do it for you. But basically we're setting, the immune spots, the mandatory fly myeloid or immune spots, tuber adherent or apparent spots, and then we're basically using those here to do our anchors, start and end, and then we create a vector of the start and end vector, and then we basically do a scalar projection of each spot onto this trajectory line. So just project along the line, and then we replot it back on to what we see. And we see a nice clean distribution, this smooth monatomic gradient that really tells us the projections were covered a general periphery to core geography. It's not noisy, so we feel great about that. And look at the pseudotime, it's kind of nice here. It just clearly, just a nice smooth gradient across, just like we would like to expect. So that's really good, really good. So where did the Banksy domains? So let's just see the pathway of the trajectory we grabbed and just saw which ones we went through. And we see here, we have this pseudotime distribution, our trajectory that goes in a very logical biological way through inflammatory myeloid to vascular lipid myeloid to stromal immune to tumor adherent. And it really tracks the biology, not just the geometry, and it kind of makes a lot of sense. And there's really an honest signature of a continuous gradient. And we feel pretty good about this is pretty good. I feel good that it's good. And then now we're looking at what are the genes that change along this pseudotime. So now we're going to look at that. We're going to get our assay, get our expression matrix, and we're going to look at them, across the line and see how they move across. You look at kind of p-values as they move across. Once again, this is kind of manual code, but it's really easy to use. You can just cut and paste it. And then our top genes rising toward the tumor, okay? And then we have our top genes rising toward the inflammatory. So the ones going toward the inflammatory are led by SPM1 TAM, SPM1, MPP1, FM1, these guys. These are all guys that are tumor-associated macrophages and CAM-ECM programs, along with myeloid genes, which was the inflammatory myeloid compartment. Makes sense. And as it moved toward the middle, it started grabbing in more things that were associated with the vascular myeloid area. And then when it moved to the abhorrent core, it switched to epithelial and proliferation. So the genes moving along make a lot of sense. And really that core signature independently reproduces 2 earlier results. The domains, heat map, and our copy number thing is actually a 3rd one. and also the GCSAs thing. So we have our domain heat map, our GCSA, and our CMB all kind of validate that too. And once again, lots of validation, great. Here are genes that are rising toward the core, so they're moving that way, right? And then these guys are kind of going the other way. You can see how they're moving the other way. And that's your kind of pseudotime for genes, right? Or these are expression, but you know what I'm talking about. So yeah, this is pretty cool. You can look over that. That seems to be working pretty well. Now we'll do a pathway analysis along that line too. Same thing. Now we grab the pathways. It's exactly the same thing. So let's say up the tumor is positive. Yeah, exactly. And then we can look at things like, EMT, genesis, hypoxia. Those looked all good. The myeloid things and things more like proliferation, E2F, MYC targets, G2M checkpoint are all things that are more in the tumor, right? Tumor core, MT. You see here, very similar to what I just said. So tumor is proliferation plus oxidative metabolism. And periphery is fibrotic stroma, hypoxia, inflammation. Both those make perfect sense, right? So the EMT lives in the periphery, not in the tumor. Hypoxia at the periphery, phox at the core. And we see it here, just nails everything. I mean, we nailed this thing. I mean, it's all making biological sense to me. All right, so we got through that. Pretty good about that. Just a really quick note on multi-sample spatial analysis. So what do you do when you have spatial samples? We talked about spatial, single-cell RNA-seq and how hard it can be to integrate them because of batch effects. Now, because the variance is very easy, it's just on the cell type, and usually your PCs capture a lot of variance early on. It's a lot easier in that aspect to integrate, just integrate the different single-cell RNA-seq methods, especially if you know your batches, right? Well, what about spatial? Now we got a huge problem, because you can't treat these cells independently at all. Like, it's not just a batch effect. Man, there's batch effects inside the first sample, not only other samples. And there's so many things go wrong when you cut that tissue and put it down in folds and all in the man, there's so many more batch effects there could be in spatial that really always exist that we just didn't know than already. So man, it's really hard to do to integrate these things. And there's a lot of methods coming out that do this. And they're mostly Python methods, deep learning methods, and they're horrible. None of them work very well. There's 10 a day that come out, they're horrible. Let's wait till maybe Claude Fable can do it. We'll wait till the next version of that, but it's not there yet. So you have to be very, very careful. And a lot of times it's just easier to compare them, to analyze them separately and then compare them. But you can't compare by clusters, right? You have to basically compare by domains. So you have to basically cluster, find similar domains, and then compare those together. I'll give you an example of how you could do that. So the expression matrix integrates the same way, it's just in a different way, right? There's different things affecting it. And coordinates aren't portable either, right? The coordinates are different. And all these side level effects, and architecture is not spatial effects. All the things I've said, it's a big freaking deal, right? So the recommended workflow is apply the same pipeline in each one, build a domain by score matrix, find the domains that look similar, and then compare those. And for example, something like this. We were able to score this. We did a z-score of looks like a bunch of different R signatures across our domains. And we would do the exact same thing to our another sample, which I'll show you in a minute in the practice problems. And then they could compare those domains, and that would tell us more about. And that's not perfect, and we'll have to just kind of wait till the better methods come along. But you'll see a lot of cool things you can compare. If you really need a unified embedding, you could do maybe harmony on Banksy PCs. You could do Banksy multi-sample with a group argument. There's also problems it's not fixing. You could do spatially aware methods, or the better ones that are coming along later. But it requires a lot more information, samples. It's not battle tested. We're still getting there, right? So right now, the score profile comparison is more defensible than integration because integration tries to get rid of batch effects. And really the big problem is it corrects too strongly. Remember how I talked about how don't even touch it if you think there's even a slight chance there's not a batch effects? You don't want to mess with it if you don't have to because they overcorrect. And imagine all the batch effects we have now is spatial. It's really going to overcorrect and be horrible. So you got to be really careful with that stuff. So now we're going to save everything here for the end. We'll give you use for the next workshop. Then we have kind of the summary I always do for you guys and a little extra stuff you can go in there to give you different thoughts and different ways you can do things. And Now we have our 3 practice problems we'll go through. We won't spend too much time since it's already been so long. I don't want to keep you, forever. Keep your attention up here, and mine too. So let's talk about just a few things, and I'll kind of go over them and give you a chance to work through them yourself. So what's problem 1? When spatial QC isn't enough. So what do we do then? I would say look to the biology, look to the H&E, right? So our data set was so clean. It had no artifacts or anything. It was just really easy. I guess that worked out pretty good to have that one and this one that's not clean. So let me show you one that's a little bit more complicated and what we would do with that if we actually happen, because reviewers want to know exactly what's going on in the H&E and if it needs to be addressed, right? So the first thing we do is look at we're looking at this, the CD429. We grab in another tumor, and what we expect to see is detached tumor fragments, tissue folding, and a necrotic region. That's what the paper told us it has. So a priori, we know this. Let's see if we can find it. So we just load up the same way we did the other one, and we look at our initial additional plots. We apply conservative plots that are very similar, exact ones we used for the other one. We don't have to worry too much about that. There doesn't seem to be that much going on with that, so it's okay to leave those, I think, pretty general. And then we do our spatial QC. We kind of take a look at everything, and we see mitochondrial focally in the corner here. That could be necrosis. And then we see this really high count here that looks like it could be a possible tissue fold. And we know there's one somewhere, and that one looks like where it should be. So let's try to think about that as a hypothesis is where that could be. So to find detached fragments, we want to see 2 physically separated pieces that have no reliable count or mito signature, right? That's the spatial. The defining factor is really geographic. You're not looking for these things. And we're going to use components analysis to kind of look for ones that could be connected. Tissue folds, these should have high in count, high feature, and you should be able to see them on the actual H&E. The pathology should be really good at grabbing those. Necrosis is often a highly elevated focal mitochondrial content with low NL feature. You can see here we have low feature here too. That's very likely necrosis. And we'll talk about how to find those. So I basically made a custom code here for the and there's things coming out in packages you can use that can do this, but I wanted to just show you kind of the behind the block version how to do this. So I'm basically looking for high in count, high in features that are anomalous and they're also vertical, because I saw a possible vertical fold. So I'm looking at building a spatial enabled graph and looking at this kind of high in count in a vertical band, and then also I'm looking for detached tissue. I'm going to use component analysis for that, and then for the necrotic spot, I want to do the local Morans, and I want to find high mito low genes. So find a spot that has high mito low genes. We can do that. Actually, I didn't use Morans here. I used a different one. But when I did all that, here's the kind of the distribution I found here. And when we plot them, here's what I got. So I got my mitochondrial right here, my necrosis, but it wasn't perfect. Specificity a little off, but nailed it pretty well. We nailed the interconnective thing, right? This is not really, I don't know if this is really torn. It might be. Maybe you should have grabbed that. But for whatever reason, it felt like this one fit better, probably. That's why I grabbed that. And then our fold, we got our fold right there. And maybe some extra one here. So there could be 2 areas here. So those are kind of areas we saw. And so our fold, we're going to filter, detach, filter, necrotic. We always, best just to annotate it, keep it there for real biology, and just remove it if needed for certain analysis. And then we'll just kind of do a sanity check here. Let's see. Oh, we filtered out everything, and we kind of looked at what they looked like before, and we lost those. You see how we lost those? We lost those. We took them out, and then that kind of just tells us about how this would be kind of a very specific clustering that we do. So that's just a way you could take on one way you can take on a harder, more difficult sample and be able to do your kind of filtering on that. So just to go back to that cross sample domain comparison we were doing between like triple negative breast cancer ER positive. Remember we made that domain plot for TNBC? Well, let's do one for ER positive now and be able to compare at the domain level. So we kind of already did this. We kind of got 4920 going up there already. We got the filtering, right? We're just going to apply the same pipeline. We just organized everything, apply the same pipeline, same clustering, same Banksy stuff. No difference. You go through it, you'll find it's just exactly the same thing. And then we ended up finding the top 8 genes for the top genes for the top 3 genes, top genes for each of the clusters. So they both found 8 domains, and then we have top genes from each one of them. And you'll see here that they found both domains, and it really just ran Banksy. If you look at this code, all we did was we basically did the exact same CRAT load, same QC. We added in the filter just for fun, and then we normalized PCA, Banksy, clustering, all the same things we did the other one, right? And then we made our domain structure, our genes per cluster, right? And then and we see that the 4465 looks right, just like we'd expect, and ER positive shows classical luminal markers, just like we'd expect. So we feel pretty good about that. So let's score the identical panels in both samples. Okay, so now we're going to do our hallmark panels, and we're going to do our add module score. And we basically, I'll kind of skim through here a little bit. We're basically just looking at these different pathways. We score them with add module, and then plot them back on the deal. And so we see 4465. Here's how we see here. EMT spread out, TLS, if you remember, T-score, hypoxia, and then we look at the same features on the ER positive. Whoa, totally different. EMT's like everywhere. TLS, pretty much nothing. Hypoxia, diffuse, T-cell, diffuse. So what are we seeing here? The TLS organization is the headline different, right? There's no plasma in 4292. So what we're seeing here is the TLS is only seen in 4465, our breast cancer, which is really interesting. TNBC has a much more diverse stromal and immune environment, right? Has more CAFs, more immune, things like that. It's very interesting. We did a positive control. Hallmark estrogen response early is higher in ER positive. That's how we expect. And we also see that there is T cells are both weak in both samples. There's kind of this exhaustion, diffuse exhaustion. So let's, now we've done kind of the comparisons there. We look at our domain scores, and remember how we made the domain for the signatures for the left, for the other one earlier? We did the same thing on the right. Then we're just gonna see which panels are the most correlated and then compare the domains. So we see things like, obviously 1 and 8 here are obviously the closest profile between samples. But there's some validity. But so we kind of gonna run a little bit of this Hungarian assignment, it's a correlation assignment to match the domains. I've shown this before, and now we see which domains kind of go and match the most, right? And for instance, we want the tumor, right? Epithelial tumor tends to be, I think, 5, right? So if you grab the epithelial tumor, the main tumor, you'll see 5 and 6. So these guys are the ones to compare for tumor. So let's compare those and see what's up with those. So we grabbed our domain scores for each one of these guys, right? Domain scores, we've already built these before, remember? When we did our plots. So we take out our scores just for tumor only and get the difference. And we just kind of look at the difference in the domains that we're seeing in the different tumors. So what does this actually show? Well, both tumor domains are epithelial positive, where TNBC is more basal-like, not non-epithelial. It has reduced liminal differentiation, and it's lost its epithelial identity in a way. Kind of things we thought, right? Yeah. And the estrogen access reaffirms ER positive. And the biggest difference is the plasma TLS signal that's in TNBC is not in the other. This is a real immune differentiator, and stromal EMT differences are more tumor domain admixture, right? So that's the way you can kind of compare these two tumors. Kind of interesting. But you're thinking about is domains are arbitrary, right? You have to make sure their numbers don't match, right? We got lucky even have the same number of domains. So it's a good way. We really have to make sure that they match. But as you can see how we're matching similar biology between two different tumors. And we could also do, pseudo-bulk aggregation between the two different ones, things like that. There's lots of things we could do to compare those once we found them. But just kind of giving you an idea of what to do, how to handle that. Final, I'll let you guys work on this one the most because there's so much, but I'm giving you the spatial module score toolkit. So remember we did that odd module score and that worked pretty well in single cell RNA, but now it's not working quite as good, but it's still working in spatial. But now we need to be aware of that, right? Be aware that it does work, but there's more drawbacks and more caveats in spatial. So be a lot more careful with it. But having an ability to make your own custom gene signature, you can't not utilize that. It's very important, right? So we're just going to run you through 5 really quick things and let you kind of just hand wave you through it to give you opportunities of ways to do things. First, build a spatial signature from your own differential genes. Well, you can just do any kind of comparison, get your differential genes, and then build a spatial signature and then plot that on there to see if you see it. So for instance, we had that tumor edge. Remember that tumor edge up for the interface? So we were like, hey, let's use those signatures, make a signature, plot it back and see if we get it. And we thought we'd get around the edges, right? But we're not really getting that, unfortunately. What we're seeing really here is the diagnostic, was it tumor versus, what did I do? Oh, I'm just going to give the edge up, right? It really just tells you that the edge up genes were not capturing really tumor invasion biology. They were capturing more this interface, this vascular stromal genes that admix into edge spots. So instead of this really pure AMT translation, we're getting more of that mixture, the bleed-in thing on there, right? And the spot, and part of this problem is that spot mixture. It's messing us up. So it'd be cool to come back to this later on when we have more resolute cell types. So we really would want to deconvolute first and then do differential analysis on really the high tumor purity ones, right? So there's other things you could do to make it a little bit better. This is an example. It didn't work that well, and that's good because we needed to know why. And it's like it didn't work. It just found more of an interaction than it did an actual edge change, right? What's another way you can use module scores? Well, how about quantitative spatial concentration with Gini? Gini is another way that looks at how correlated things can be, and we ended up just grabbing different hallmarks, and we scored for Gini instead of at module score for this one, and we looked at Gini, and we saw we kind of looked at we kind of saw what it was, and it wasn't super great. We decided to compare that. So we're gonna have Moran's eye and Gini, right? So a high if they're both high, we have focal structure. If they're both low, they're diffuse, right? And we kind of saw what we got, and we really see a interesting split between the two of these, where we have more spatial structure, more of the Moran spatial structure here, and then more of the Gini. So I guess the Ginis always, for some reason, have pretty low facial structure for whatever reason. But they have comparable high concentration for those. So there's really just different ways. We basically got diffuse, random, and broad structures, two things. And it's basically just part of this is the property of the module scores, not the tissue. Just averaging the genes, things like that, and comparing its problem. But here's some kind of patterns you can look at that are kind of very interesting. That will teach you a little bit more about the biology and kind of inform a little bit more. And the bottom line is Moran's separates structured from random, where Gini's ranks within tissue concentration relative to other modules in the same tissue. Right? These are different things for different reasons, but another wrinkle in ways you can tackle this to be able to pull out information. Okay, one more place you can do a spatial co-localization between pathway pairs. So basically you can score 2 pathways and be like, I want to see if these pathways, just tell me if these pathways co-localize, right? So we basically do a Pearson correlation, or you could do even local Morans, and you compare if they are correlated, and we see not super great. And we use then we compare that to Lee's L's, which is a spatially aware version of the co-localization, and we see the difference in those and see how they compare. And we kind of visualize the strongest one, the hypoxia epithelial MMT one, and what's the result. 3 of the 4 bested pairs show meaningful correlation. 3 of the 4 are okay, but the canonical TMBC prediction that we want, the exhaustion plus M2, kind of failed. So these kind of show TMBC is kind of shows a bit weak. So, but really, why did that one fail? It's because of the diffuse and random, right? These two scores are very diffuse and random, and the scattered, they can't co-localize enough. So it's not that this isn't immunosuppression, right? That it is happening. It's just more diffuse than localized. You can read a little bit more here about Liesel versus Pearson here. All you do is ROI level. This is what you do in DSP, right? Well, you can just grab little tiny regions, like I mentioned before. You can grab a region that has a lot of pollution in a LUAD sample compared to one that doesn't. You can grab a bunch of regions that have pollution and compare them to a lot of ones that doesn't, right? So the point is you can choose ROIs and compare them. Now, remember when we compared different domains, we were worried about pseudo-replication using find-all markers? Well, that's a real problem. And it wasn't really a problem there because all we cared about is log2 fold ranking the cells. But here, this is going to be a much bigger problem because we're actually really caring about the biology of different spots. So we cannot do the find-all markers again. It's going to inflate and cause pseudo-replication. The better way to do it would be to aggregate the spots, either compare the domains themselves or aggregate the spots themselves, either with pseudo-aggregates or just Wilcox together with those spots. That's the only way you'll get the true right, the right scores that actually matter, right? You'll see here, if you do a spot level test, it totally inflates the p-values. If you do an ROI test, we're just doing the plasma score in tumor versus non-tumor. That's it. So this is showing we have a huge correlation. This is showing we don't. So the idea is, there's differences are similar in magnitude and direction. It just matters how you're asking it and what you're saying. But it just shows the problem of pseudo-replication, how important it is to not use that find-all marker thing. You could use find-all markers inside a single cell sample because they're all the same at that instance. We're just working on the PCs. But if you do it on the artifacts of the spatial, it throws it all off, and you can't do that. One more you can do, you can translate a published signature. So you just go out and find a really cool published signature that fits your data that might lead to a hypothesis on clinical outcomes, something like that. I don't know, anything, right? So we pulled in this melanoma T-cell exhaustion signature, right? And wanted to see how well it would work on this TMBC thing, right? So we pulled it in, grabbed the signature. You could have grabbed anything. I module scored it right, checked it. Let me see kind of the diffuse. Doesn't look really that great, to be honest with you. The Spearman between exhaustion T cells 0.424. We kind of look a little bit further, and really the exhaustion signature kind of partially fails. We're really measuring more T cell abundance, not exhaustion state. So what's the way to do this a little bit more controlled? Okay, that was a hard thing to look for. Let's look at CAVs instead, right? So CAVs marks a cell type, not a cell state. In this instance, maybe you'll be a better controlled thing. So now we grab this CAF signature, do the same thing, and now we see kind of more interesting signature, I think, to where the ECMs and stromal areas really are. But anyway, you can kind of look into, the idea is you can use anything. That's the main thing, right? You can use anything. Any kind of custom score for add module or any kind of Gini, any kind of thing. Think about ways that you can apply to the spot level, especially when they're deconvoluted, to be able to visualize these cool patterns within your data. That's what makes it cool, right? Here's kind of just summary of our practice problems and then some key takeaways, like I always do, common pitfalls, additional resources, and that's it, workshop complete. I think we did a great job. We started out with our raw accounts, did our filtering, normalization, we did principled, we did really great outlier detection. We did our clustering for domains and annotated the domains, validated with copy number inference. And we learned a lot about, applying, different cell type scores, domain analysis, trajectory analysis, everything. So the next step is we're going to do all these things. The next step is we're going to do deconvolution to get the actual real cell types, because we want to. Those cell types are mixtures, right, in spots. We want to know what's going on in the actual spot. What's the cell type in that spot? Not is it just the biology looking like it, right? So that's going to be very important. And then we're going to actually apply a very similar downstream analysis after that and take it further. And then we'll see what we got from there. So I think it's going to be a great, fruitful workshop. I hope you got a lot out of this. I had a great time going over it with you, and Ready to see you guys, and I hope to see you for the next lecture. See you then.